Hi, good afternoon, everyone. I'm Rajkumar Agarwal, representing Veeda Clinical. It is my privilege to offer a heartfelt welcome to each and every one of you today. We are delighted to have you all here for a symposium on integrated phase 1 trial solutions for emerging biopharmaceuticals, addressing development challenges and accelerating timelines. We have organized this event in collaboration with Atal Incubation Center, and this collaborative initiative aims to address the challenges and optimize the process related to the development of new chemical entities and large molecules. Covering various aspects within these domains, we have gathered an illustrious panel of experts from the pharma and biopharma industry, researchers, scientists, and esteemed regulatory experts. The symposium aims to provide a platform for sharing valuable insights, experiences, and best practices. This information exchange can contribute to the collective understanding of how to navigate the complexities of developing new pharmaceutical and biopharmaceutical products. We also extend a warm welcome to our virtual audience who is joining us from the various parts of the world. Without any further delay, I would like to invite my colleague Yuvragi to take it forward. Thank you.
Thank you, Raj, and a very warm welcome to all our esteemed guests. This is Yuvragi, and I represent Veeda Clinical Research. I'm extremely privileged to introduce Dr. Madhusudana Rao, who will be delivering the opening remarks to welcome our virtual participants, guests, and fellow speakers. Dr. Rao serves as the Chief Executive Officer of the Atal Incubation Center, CCMB. He holds a BSc in Agriculture from AP Agriculture University, Hyderabad, followed by an MSc in Plant Breeding and Genetics from Punjab Agricultural University. Dr. Rao's academic journey culminated with a PhD in biochemistry from Osmania University, Hyderabad, which he completed in 1987. With a solid educational foundation and extensive experience, he now plays a pivotal role in fostering innovation and entrepreneurship at the intersection of biology and technology. Please join me in welcoming Dr. Rao on stage. Over to you.
Good afternoon, and on my personal behalf as the head of the Atal Incubation Center at CCMB, I welcome each one of you to be here, and especially the dignitaries and the sponsors for having taken such a trouble to be here and then arrange this very important meeting. And if you look at the title, in fact, this is one of the bigger, one of the kind of often seen obstacle in progressing from a proof of concept to the kind of a product stage. A lot of times what happens is there is very little advice to startups who are very green behind the ears in terms of how to take their idea or a product to the market. There's a lot of mentoring is required. And of course, there is a lot of help that comes from the DCGI, CDCSO and things like that. But at the same time, the engagement and the interaction has to be much more for these things to happen. And entities like Veeda and things like that, they are experts in performing these clinical trials. But again, they have to have an interaction with startups who are developing these interesting molecules that may have a value. So when briefly about our place, Atal Incubation Center is an incubation center for life science startups. Proposed and established by CCMB. As you know, CCMB is a national institute in one of the labs of CSIR and focused on health, fundamental biology. So our Atal Incubation Center also looks at supporting startups which are in life sciences and biotechnology. So when I see in last 6 years, we have been engaged with more than 100 startups who are in health sciences. And we are fairly successful in graduating quite a few of them. And they went out and raised a lot of money. So the point here is, such an activity is very important. As you know, India is a very well-known knowledge center, knowledge generation center, but at the same time, when it comes to translating this knowledge into some kind of a product or a process, we are not there. The reason for this kind of a thing this gap to happen is the enabling entities are less. But in the last few years, we see a lot of enabling centers like, these startups, incubators and things like that are able to bring in the developers with the regulators and various other entities. And I think in that context, this particular meeting and getting together is extremely important for us. In fact, if you look at the kind of facilities that are going to come up in Hyderabad the next few years, are mainly to support the transition from proof of concept to a next stage. Like, how to make a 3 identical batches. If I go to a private player for a startup, it costs more than 15 lakhs. They can't afford, the thing is still at a developing stage and still at he doesn't have enough strength to raise that kind of money. So we need a lot of support systems that come from the government and other entities to make this journey much more easier, so that there are more NCEs, new chemical entities come from India. In fact, we don't want to be called as follow-on biologic country. We've been there as generic people for a long time. And there is no reason why we can't be a new molecule entity country. So all this is because the processes need to be much more addressable, much more easy to accessible and things like that. I think this particular conference, where we have a very distinguished guest from ICMR and also from the DCJ Telangana and the Veeda people, I think this is a great opportunity for all of us. And we also want to play our little role of enablers in bringing these people together and make things happen. This is our thing. And, today we may meet for a couple of hours and leave, but I welcome each one of you to come back again. And then, if you have anything, we have them in contact, and we can, any help, please don't hesitate to ask. If you go to any conference these days, they'll tell you one thing: everybody is working in their own silos. There has to be much more interaction that need to happen for people to, have clarity on many matters. So I hope next couple of hours will be a good time for all of us to understand the landscape of regulatory issues, and we have excellent experts and have been there for a long time, and I'm sure their advice would be very, very valuable. And thank you very much for being here. Thank you.
Thank you, Dr. Madhusudan, for your wonderful opening remarks. Next, I would like to introduce you all to Dr. Ramji, whose contributions have been instrumental in making this event possible. Dr. Ramji currently working as Chief Operating Officer at the AICCMB, Hyderabad. He has a rich background in biotechnology and innovation. Prior to this role, he managed the big scheme at IKP Knowledge Park. Dr. Ramji has over 40 research publications, and he is actively involved in advisory roles for research and academic institutions. He also serves as a governing board member of ISBA and a board of directors member of NIPER Hyderabad, showcasing his commitment to advancing biotechnology. Please join me in extending a warm welcome to Dr. Ramji to deliver his opening remarks for the symposium.
Science today. So, Madhu and me, so we gave some positive science. So anyway, thank you, all of you. Good afternoon to each one of you. As Madhu started this AICCMB about 6.5 years before, every scientist and student probably, thinks that, it's not our cup of tea to join this kind of entrepreneurship activities and the programs that we are doing. But, as we see the journey here, a couple of students start the companies, and a few of the scientists also started the companies. But yes, we created the ecosystem, not only in the state, but also in the country for startups. If you can see 0.1 million startups today means it's not a joke. So pandemic times and, post-pandemic, a lot of changes that happened. But, we are good in science, innovation, and kind of small translations. But coming to the enterprises, and again, scientific enterprises, we are a bit lagging behind. So that's where we're trying to actually fill the gap by connecting certain dots between various ecosystem partners. If you can see the landscapes, briefly, for any startup or a scientific enterprise or any innovator to think about starting a business, it's like IP landscape, market landscape, competitors landscape, and most importantly, the regulatory landscape. The past 10 years, when I worked with my previous organization and also here, we used to connect to the regulatory agencies or some of the CROs. We have a startup like this, they're developing some device. Can you please help them? So the challenge here is, as I'm talking to Ramkrishnan sir, right approach to say, this is needed, this is not needed. Who is going to draw the line? We are stuck here. Most of the incubators, most of the enablers in the ecosystem, we have startups, we have scientists, we have wonderful ideas, technologies. But when you're trying to go for a scale up and the regulatory funding also, of course, slightly improving, not from the government. Government funding is good. The private funding I'm talking about, the VCs and angels coming into the deep tech innovations like this. So who's going to draw the line? Is there a single window mechanism from the local CDSCO or the central CDSCO where, boss, this is the thing. These are the agencies, marked agencies you can follow. Or maybe some of the guidelines or quick guidelines, this can be done. Maybe 2-3 certifications are only needed, not 4-5, because most of these regulatory clearances can be done by the government support. The government money that they get is very limited. It's about 10-15 lakhs in a BIG grant, or maybe if seed fund or maybe any other grants that they get, maybe about 30-40 lakhs they may be having, right? How to actually minimize those costs when they are actually trying to go for the regulatory. So I think just to start a dialogue today, the initiative by Veeda, I think Kiran sir, actually we met in last year, December or sometime. These are the challenges if you could able to address to our startups that, how to actually approach to a regulatory agent, okay, who can guide you. Like these are the 2-3 steps followed. Not all certifications are needed for this device. That is the problem here. If you are going for going to spend some 3 certifications which are enough to clear your regulatory pathway, you will be saving some money and time of the innovator and the government money also, or maybe personal money, whatever. I think down the line, sir, I think, this kind of advisory is very much important for our startups. And we are very fortunate to have this collaboration with Veeda, Dr. Kiran and the team, entire team, and of course the local CDSCO, directories here. And we invited Dr. Suchita also from ICMR. Unfortunately, US FDA guys couldn't join even for 10 to 15 minutes, virtually asked them to join, but they are flying today. So otherwise, we'll be doing more such kind of events, but please enjoy these 3 to 4 hours of engagement with excellent line of speakers. And I welcome you all once again to AIC CCMB. Thank you so much.
Thank you, Dr. Ramji, for your opening remarks. We are now about to commence our engaging sessions, and I have the pleasure of introducing our very first speaker and our chief guest for today's event, Dr. A. Ramkishan. Dr. Ramkishan brings an illustrious 25 years of career in regulatory services at the Central Drug Standard Control Organization under India's Ministry of Health and Family Welfare. He served as the head of Biological Division at CDSCO, overseeing the biological product reviews and clinical trial approvals. Additionally, he managed responsibilities related to manufacturing licenses, import regulations, and more. Dr. Ramkishan has international recognition, serving as a member of the World Health Organization's Expert Advisory Panel on Biological Standardization. His contributions have been acknowledged with numerous awards, including the Dr. B.V. Patel Gold Medal and FDA Person of the Year Award, reflecting his exceptional work in the regulatory affairs. He also served as an expert committee member of Indian Pharmacopoeia Commission, National AEFI Committee on Vaccines, and more. So please join me in welcoming Dr. Ramkishan to begin his presentation. Also, I would like to invite Dr. Madhusudan to honor Dr. Ramkishan by presenting a bouquet.
Okay. Respected Dr. Marsoon Raoji, Chief Executive Officer, Atal Incubation Center. Respected Dr. Ramji, a pure scientist, taking care of a lot of important initiatives in the area of drug development. The main person behind the show for inviting me here and to connect all scientists through our Ramji and Marsoon Rao sir, Dr. Kiran Martakji, Director, Regulatory Affairs, working in Veeda Clinical Research Services Private Limited, near to AMC, Ahmedabad, Gujarat, India, and the pure scientist, Dr. Jagannath Kota, who is going to speak on the biomarkers in the clinical trials, and respected Suchitra Madam, the pure scientist from ICMR, with respect to strengthening the ecosystems on clinical trials and important medical devices and other drug developments, Dr. Raviji, and one more important speaker, our Bharadwajji, and all other eminent speakers and invited scientists and startup captains, regulatory friends from my office, Dr. Shavan and Dr. Sinhaji. He is a director heading medical affairs and clinical research from Hetero Drugs Limited. And ladies and gentlemen, good afternoon to one and all. Yes, what Ramji sir is told is 100% correct. The main challenge in our country is the regulatory, industry, academia interface is one of the big challenges. So on behalf of entire Central Drug Standard Control Organization, on behalf of my respected boss, Dr. Rajiv Singh Raghuvamshi ji, who is one of the renowned pharmaceutical scientist, the apex body regulator heading our country. So with his blessings, we have visited at IICT 4 or 5 months back in the month of May. At that time, I was little passionate to play the badminton. At that time, I fell down. Today, I'm sorry you fell down, sir. Some connection. So God is so kind to connect all of us. So scientists are always unorganized. Others, I don't want to comment, they are well organized. So with this, definitely you will enjoy for 20-30 minutes what are the expectations from the regulatory agencies with respect to your novel new chemical entity developing and empowering with respect to submitting the important data on safety, purity, efficacy, and other toxicokinetics. So toxicology also we are covering. That because no, as a regulator, our main objective is to protect and promote public health is one of our vision. Therefore, whatever drugs are being approved, cosmetics, medical devices, in vitro diagnostic kits, as per the new medical device rules 2017 and as per the our revised schedule M rules because no 1988, 1992 and 2001, 2006 and 2019 the new drugs clinical trial rules framed as per the directions of Honorable Supreme Court of India, the new GSR 227 E dated 19th March 2019. We are with a well-defined, comprehensive regulatory guiding principles for promotion of new startup companies like you and the promotion of clinical research. I can say, I don't want to call good clinical practice. Just yesterday, we had a discussion with the US FDA, Dr. Robert Califf and Dr. Sarah Countryhead. I was also one of the pure pharmacologists attending off the dais. So we raised it when the Indian market is $1.93 billion market, CAG at the rate of 8.5% when we are taking care of the growth rate, and when India is contributing out of $90 billion market of ANDA submission to USA, only 10 billion we are contributing. So now we talk digitalization, automation with respect to carrying out all production operations and control step by step, starting from receipt of raw material to our primary packing is completed, then followed by the finished goods storage area till it goes to market and prescribing to various patients as per the prescriptions of medical doctors, physicians to achieve optimal health outcomes in hospital and community settings is the important thing one should note. So friends, you are all scientists. Once molecule is identified, for example, para-acetaminophenol. So you are having ring, molecular weight, melting point, physicochemical characteristics, solubility, all important other description, then development starts, then stability, then method validation, then stability. Then again you have to prove whatever, just I'm setting an example. Then proving safety and efficacy in the animal models. Then you have to carry out again human trials once the safety and efficacy along with our mutagenicity, reprotoxicity and other toxicokinetic parameters as per the regulatory guidelines you have to submit. Then definitely now Government of India under the able leadership of our Honorable Prime Minister of India, Narendra Bhai Damodar Das Modi ji, and under him our respected Dr. Mansukh Mandavia ji is taking Honorable Health Minister, Government of India, taking a lot of initiatives with respect to promotion of industrialization in order to make sure that the products whatever being produced under the GMP environment to make sure that all the available drugs are meeting predetermined standards as specified in various international as well as our Indian pharmacopoeia or in-house specifications to ensure patient-centric effects is the important object. This is number 2. Madam Suchitra, हम आपके साथ मिलेंगे. Now things are changing. The regulatory mindset is changing. We are also working like an enabler, facilitator. Olden days gone. At the federal level, you can ensure since May 29th, 2014 to till date. Now again we are due for elections. So things are changing. When we are having excellent talent pool, excellent scientific pool, excellent demographic conditions, fast growing economic conditions, then where we are lacking is the question. When because of all these important molecules, generics, we are number 1, almost 4,502 drug master file filings by India. $6,585 billion market ANDA means approximately filing approval 6,585 companies approved for ANDA molecule contributing $90 billion. India is contributing only 10 billion. In 2002, 2022, despite of our post-COVID challenges, our pandemic situation, we got 101 ANDA approvals through remote monitoring, through virtual interactions, through videography. So things are changing, friends. हम कोई किसी से कम नहीं है. We are better than USFDA. Only thing is we have to revamp. How the ICMR people took key role during our pandemic situation with respect to approval of all important in vitro diagnostic kits. 73 IVD kits we have approved, domestic as well as imported kits, and we have validated with respect to specificity, quantification, reactivity, sensitivity to meet at least 98.22, 99.9999 something. Efforts made by all startup companies like you and good companies like other important multinational companies also kept their efforts, but where we are also changing. 48,500 ventilators we have audited across the country. As a regulator, हम भी ventilator पे जाने वाला था, बच गया. So God is so kind, and thoughts are changing, and we have to put our thoughts out of the box, making allegation, wasting valuable energies and time. Those days gone. My appeal to all young talent minds sitting off the dais, please concentrate. Sky is the limit. God will bless you. We are all with you to facilitate. I am heading Andhra Pradesh and Telangana. 16 months में हमको पहला invitation आया. Always I go since 1987 to आज तक हम हमेशा जाते हैं. I belong to Warangal. I have referred our CCMB IICT library in the year 1989 when I was in graduation pharmacy. I am having high regards, and Lalji Singh Saab, 3 times I got the opportunity to interact with him, and when he got the vice chancellorship in Banaras Hindu University, Varanasi, and he was the chairman constituted by Government of India to revamp the stem cells product guidelines. I was the member secretary, and Balram Bhargav Saab, हमारा सौम्या स्वामीनाथन मैडम, VM कटल साहब, YK गुप्ता जी, मेहरा साहब, हमारा और एक मैडम है, 2-3 विजय कुमार, 2-3 मैडम. So we are always proactive in those days when I was heading biological product. At least you should understand, I am not exaggerating myself, but ये self डब्बा बोलते हैं ना, ऐसा नहीं है. So one should understand what are the gaps we are having. रामजी साहब हमको वो पेन हो रहा था. He was one of the participants of the day. So we have not compromised. That's what I want to convey, friends. So think positively. We are all here to facilitate and support our Masoon Rao Saab and Hamara Ramji with respect to your speedy approvals. So on behalf of CDSO, I definitely assure you, come and visit our office, go through the website www.cdso.nsa.in. At least you can have all important guidance documents, checklists. Because we are not expert in preclinical, frankly speaking, because I'm a pharmacologist. I know the animal models, how to give the STZ with respect to rats to develop the diabetes, because my research topic was the development of new drug formulations of insulin with the trace elements of proven anti-diabetic activity. 16 weeks ka hum study kiya. Fortunately, my thesis went to Professor John McNeil, Department of Clinical Pharmacology, Toxicology, Canada, Winnipeg. So hum aisa kuch nahi hai. We are also having 50 publications. We are also putting efforts. So please, maybe we are not like a developed country like USFDA, but with limited resources, with limited knowledge, with limited exposure, around the clock we are putting efforts in order to promote industrialization under Make in India initiative, under ease of doing business. 24/7 we are working in customs offices. I worked in Bombay, Delhi, Gujarat, Calcutta. After 24 years, I got the opportunity to work in Hyderabad. I never expected to come here because I like Delhi. Once you work in Delhi, east, west, north, south block, south block, bahut hai. So, Suchitra madam can understand. With this, I'll go through the regulatory requirements with respect to clinical trial. And when you are engaged with the biological product in the year 2016 and 2012, we were having two guidance documents. 2012, with the support of DBT, Department of Biotechnology, S.R. Rao Saab and Vijay Raghavan Saab was the secretary. And I was the convener, and Dr. Vijay Somani, myself, we used to closely interact with the DBT and developed a comprehensive guideline for market authorization on biosimilar products in India, which was established and published in the year October 2016. First time biosimilar guideline, biosimilar definition, we have incorporated in that guidance document. Sample size, we have kept in that. And important critical key quality attributes, critical quality attributes, we have mentioned in that guidelines. So we are also putting efforts in order to have the alignment with respect to ICH guidelines, WHO guidelines, EMA guidelines, PMDA Japan guidelines, KFDA Korea, US FDA and UK MHRA and other guidelines. At the same time, WHO in the year 2022, May 22nd, 75.8 resolution passed in order to strengthen the clinical trial environment, clinical trial ecosystem. Strengthening on clinical trials is very clear. Now the draft guidelines published for your comments. Please give maximum comments from Indian side so that next month I am going, I got the opportunity to serve in the Biological Standardization Committee headed by Dr. Ivana under the Director General of WHO Geneva. And first person, I am the only one person from the entire country, madam, nominated me as the expert advisory panel based on my publication, based on my technical, it's not as a regulator. Godfather, based on the virtual interaction through 2-3 series of the scientific discussion. Based on that, God has given me the opportunity how to reduce 3R, replace, refine, and reduce and alternative models with respect to the animal models available. So guidance document is ready. Definitely, I would like to request Ramji to give your thoughts so that we can incorporate in our upcoming the guidance document by WHO Geneva. So with this, thank you very much, madam. And I am not that great, but as a pharmacologist come regulator, I will put my efforts and what are the important principles involved with respect to the Indian drug regulatory framework with respect to ensuring quality, purity, safety, efficacy of drugs to achieve patient safety in clinical practice is one of the important topics, friends. So these are all the important contents of my outline of the presentation. The regulation, because now we are having 103 rules in our NDCT rules, 8 schedule, 13 chapters. How many people read that guidance document? Can you raise the hands? बहुत busy रहते. We are all busy with the Google search. रात को 2 बजे को सोते, हम आने वाले समय हमें शादी करने की जरूरत नहीं है. But please, my appeal to all young talent scientists like you, please go through the guidelines so that you can understand. 103 rules, 13 chapters, 8 schedules, and various forms, city 1 to city 37. So city 1, 2, 3 application fees and all, I will show it to you so that you can understand. And the main principle involved in our Indian drug legislation is the main law enacted, which was enacted in the year 1940, Drugs and Cosmetic Act. One should know, this is our Bhagavad Gita as far as our Indian regulatory system is concerned. And the rules framed in the year 1945, we call Drugs and Cosmetic Act 1940, rules thereunder 1945. This everyone knows. But why? Why drug inspector should come? Why pharma? हम तो डॉक्टर है, डॉक्टर को ज्यादा मालूम है, फार्मासिस्ट क्या करते हैं? ये law है. This is the law. We are all regulating blood centers, blood products, vaccines, recombinant products, cell therapy, gene therapy, tissue engineering products, allopathic medicine, phytopharmaceutical, CROs. ये सब law में है. So drugs are very, very important. Drug is nothing but any substance, any liquid item or important molecule meant for diagnosis, mitigation, cure or prevention in human beings or animal elements either for internal or external application is considered as a drug. So as per the section 3B of Drugs and Cosmetic Act, it's very clear. Whenever you go for the development of any molecule, if it attracts the words what I told with respect to definition of the drug, then it is called as a drug. Simple. And the devices, MDR rules are very clear. Any apparatus, any appliance, any material, including software, meant for same diagnosis, mitigation, elevation, cure in human beings or animal ailments considered as a medical devices as per the medical devices rules 2017 based on the Global Harmonization Task Force GHTF consisting of TGA Australia, Health Canada, European Union and USA. So we are not compromising. Only thing is implementation is the challenge. Interpretation of the law, understanding of principles is the challenge. We always consult our consultant. They always confuse you and confuse us. They will take their own advantage and they will do the dhanda, what we call business. So don't trust anyone. As a scientist, you can come to our office. We will guide you. We will facilitate you, sir. So there are all the things. So this is the background information with respect to the DNC Act. Main preamble of the DNC Act is to take care of the regulation of the manufacturing operations and control, distribution, sale and import of drugs as per the chapter 3 of Drugs and Cosmetic Act. For all devices, medical products, we don't have any term as medical products. We call in our country drugs, cosmetics, IVDs, etc. So the objective is DNC Act is to ensure whatever drugs are being produced in different manufacturing facilities, whatever public is received, it should have proper safety and efficacy, ability to interact with the tissue proteins called as receptor, drug receptor complex once it is developed to elicit a desired pharmacological effect to treat any disease or disorder in healthcare system is nothing but a important our patient safety. So this is as per the section 3B and basic philosophy everyone knows. I don't want to hammer. I will give all 45 slides, but important applications it is more useful for you, scientists like you. So research. So good because clinical trials में हम CRO में गया good clinical practices बोलते हैं. कल USFDA बोलते why good clinical batching what? So this is the research, academic research, observational research, clinical research, regulatory research, translation research, analytical research, and bioanalytical research, and other important parameters influencing the overall research, overall drug development is concerned. So you can take care. So research in the area of drug discovery is a very important tool, friends, to ensure the availability of the NCEs, new chemical entities, available to the public, vulnerable population with proven efficacy and safety, you will get drug approval in order to have your market authorization, sale and distribution moving in international commerce, friends. Clinical trials is absolutely necessary to establish the scientific data. Unless you have the proven safety and proven efficacy, how FDA will give the approval? There are set of guidelines, principles, methodologies incorporating the regulatory guidelines. So one has to stick to that and you have to submit the reliable data with scientific evidences. Then only you will get the approval, otherwise no. So what are the clinical trials, friends? Everyone knows it is a systematic study of novel NCEs in human beings with proven data, with scientifically proven data and authenticity, archival and the legible form formats, including your electronic case report forms and other data is available. Now, after 2-3 years, we are going to have complete digitalization, friends. Electronic manufacturing records, electronic batch manufacturing records. Totally, globe में 70 companies going through the complete digitalization. Out of 70 companies at the international level, 2 companies belong to India. One is Cipla Limited, Mumbai. Second one is Dr. Reddy's, Hyderabad. These are the 2 companies as and date complete distillation mode. So As far as CDSO is concerned, 90% we have geared up with e-governance. Complete online application. We are not accepting offline information. So things are changing. Accordingly, you have to gear up and you have to prepare with respect to your futuristic submission as far as the drug approvals are concerned, friends. Then it is of paramount importance that all clinical trials, because here, whatever subjects you have involved with respect to the clinical trial process, starting from phase 1 to phase 4, how you are going to ensure. ICMR came with well-defined guidelines with respect to biomedical health research, with respect to the participants, human healthy volunteers, human subjects, sometimes patients in novel cytotoxic molecules during your BAB studies or clinical trials. So now we are having set of guidelines in order to protect, promote their well-beings and rights and safety of the people, those who involved with respect to the clinical trials, BAB studies, friends. So this is one of the things I want to just highlight you people. And the trial subjects are protected and data generated should be scientifically valid. Clinical trial of new chemical entities be initiated only after obtaining the permission from the Central Licensing Authority. Please don't go for violation. Last month, one doctor in Mumbai, without taking permission, he started the clinical trial and his site was barred, debarred and blacklisted. 2 minutes अभी हमको WhatsApp आ गया, 2 मिनट में पूरा वर्ल्ड में चले जाएगा. Law is in place. He is a learned personality and carried out on his own. So should we expect that he is a layman? I am not criticizing, I am telling the truth. So please, law is one, because we have to respect. Everyone has to respect law in our country. So friends, don't do any violation. We are always here to facilitate with respect to that. So licensing authority with respect to clinical trials, approval of new drugs, approval of vaccines, approval of test license with respect to investigational molecules, new chemical entities as per the rule 2W, where it deals with investigation new drug. Earlier we used to discuss under rule 122E of Drugs and Cosmetic Act with respect to new drug definition. Now as per the NDCT rules of 2019, rule 2W, rule 2S, investigation new drug and new chemical entities, elaborated and including phytopharmaceutical. I will give the definition of that also, friends. So these are all the important phases of the clinical trials. You are all doctors and scientists. I don't want to hammer, but yes, these are the phases of clinical trial with respect to proving your safety and your dosage, and important your PK/PD parameters and efficacy with respect to the novel drug development process concern. Sometimes after completion of phase 3 clinical trial, you are going to have waiver for the structured phase 4 clinical trial. But sometimes if very few subjects involved, some orphan drugs or important categories of drugs, we are going to approve the market authorization with the subject to the safety information data generated with respect to condition to carry out the phase 4 clinical trial also. अच्छा, नहीं दिख रहे सर? क्या क्या? अच्छा, अच्छा bold है सर, थोड़ा देखो सर. Because of this light maybe. देखो देखो. So all these are very very important. नहीं मैं सर without light अगर 3 घंटा बात करें मैडम को देखे डर ही लगता है. So because we are all creative scientists, so one should not be of India, sir. So based on the Union of India and Indore Chacha Nehru Hospital in the year 2011-12, I was the investigating agent with respect to the Bhopal victims, right? So, sir, hum with limited manpower, doctors are putting efforts because of our 125 pages report. Supreme Court appreciated and gave the directives where we are having now NDCT rules of 2019. We are not claiming, Ramkishan is not claiming. But this credit goes to all my regulatory friends, my DCJ sir, Health Ministry official, Honorable Prime Minister of India. So we never claim, sir. So low profile, visionary with respect to the global environment is concerned to have fast track approval. So if you see the how the city regulations evaluated, so that I would like to take care here. So in the year 1988, I already told, sir, 1988, 84, 94, 96, 2005, and February 2013. So just I'll go through that. I will give the presentation copy to you, Doctor. Don't worry. You give your email. I will send it to you. Don't worry for that. You are from which place, sir? You are from CCM, sir. CCM means Center for Cellular. C means commitment. Again, C means a different. Huh? Don't worry. So when I entered into this, I was observing the light. More lux value leads to the dullness with respect to retina, where you can have sedation or boring. Third, lack of interest हो जाएगा. तो आराम से last bench वाले को नींद ही आ जाएगा. I know. But with limited energy, I am putting efforts. Try to be in alert mode so that at least they can have few slides where they can have some important good principles they can incorporate it in your left brain. Left brain takes care of the innovation. Right brain takes care of the creativity. Anatomy, if you can go through, 1200 grams, gentlemen, adult human beings are having, and 1300 grams, all ladies. So, women's, respected women's in our country, 100 grams extra. So, our Honorable Prime Minister of India really identified to have 33% of reservation. I don't know, sir. So, as a anatomy padhai karke bol raha hu, sir. Just I am sharing for curiosity. Now it is okay, sir? No. Okay. Okay, sir. So these are all things, Doctor. So in the year 1988, first Schedule Y came, sir. Actually, Drug Act came in the year 1940, sir. Cosmetic regulation in the year 1962. Then 1979, there was a draft. Then 1988, this first Schedule Y draft came where it deals with the preclinical and important human pharmacology, safety and efficacy parameters involved with respect to human pharmacological aspects like pharmacokinetics, pharmacodynamics, mechanism of action, dosage and dosage form, including contraindication, side effect, Doctor. So 1994 TRIPS agreement that you know, Doctor. 2000 May we got well-defined guidance document by ICMR Delhi with respect to biomedical health research, Doctor. Now, as per the NDCT rule, chapter 5, we are having a well-defined chapter, separate chapter. Out of 13 chapters, 8 schedules, 103. आप बोलो, आप एक बार देख सकते हो if I am not wrong. So you can correct also. So you go through that academic clinical trial, biomedical health research controlled by ICMR based on the ethics committee permission, Doctor. Those who want to go for the regulatory trial, they have to approach our DCGI, Drugs Licensing Authority. So in our country, we are having institutional ethics committee and independent ethics committee. Almost in our country, we are having 1,766 ethics committee permission registration granted by our Honorable DCJ, sir, Delhi. In that, layman, basic scientist, important pharmacologist, one clinician, one lawyer, one NGO, and first time we have incorporated one representative from women's side. Ladies representative. I made a, sir, sab log gents raha to kaisa? Equity of justice. First time in our NDCT rules, ethics committee composition, go through that, we have incorporated one women representative also. So this is the background and the regulation before 2009, before 19 March 2019, clinical trials were regulated under Part 10A of Drugs and Cosmetic Rules and Schedule Y of the Drugs and Cosmetic Act, sir. Now we are having GSR 227E published on 19th March 2019, Doctor, that I will tell you, Doctor. Here, how we got this? Based on ICH E6, E8, we have developed important guidance document for clinical trials. And World Health Assembly, already I told, resolution passed in the year 2022 with respect to 75.8. Jagannath Sahab already understood. So every country is having their own set of guidelines with respect to approval of clinical trials, sir. And these are all the concerns during the 2010 and 2012, 2011. I have done the audit at Bhopal Hospital, Indore Chacha Nehru Medical College, Indore, Madhya Pradesh. So, ethics committee review, we were not having effectively. Informed consent process, wardens and other people are doing signatures. Compensation, no compensation, those who participated in the clinical trial in case of death or serious adverse reactions, including some vital organ failures. Then compensation for accidental injury, we were not having, and review of clinical trial inspections, we have not geared up. As a pharmacologist, this is my passion, so at least I can commend you, sir. And analysis of serious adverse event. Now we are having separate casualty committee with respect to SAEs as per the Indian Pharmacopoeia Commission recommendation. If drug regulatory actions required, we are taking care with respect to compensation as per the casualty committee. Nowhere in the world having this type of provision with respect to compensation formula in case of death and in case of accidental injury, in case of medical management. Suchitra madam knows very well. So with the support of ICMR, we are taking care and we want to give the good message, eco-friendly environment with respect to regulatory approvals, friends. So these are all the major amendments in 2013 took place, GSR 53, 2013 and 2013 again 63 with respect to conditions for the conduct of clinical trial, authority for clinical trial inspection. So whenever we go for the clinical trial inspection, we are involving few subject expert pharmacologists from different approved medical institutions and reputed medical hospitals, important civil hospitals. So we are taking few people along with our inspection team to carry out effective clinical trial audits, sir. So this is also one of the important oversight. Clinical trial oversight is one of the major indicator in the national regulatory assessment by WHO Geneva. WHO Geneva, along with few other regulatory countries, experts, 15-20 people will come to Delhi. They will do the assessment. What is our national health policy? What are the inspections we are taking care across the country? How effectively we are carrying out as per the provisions of Drugs and Cosmetic Act and NDCT rules with respect to implementing good clinical practices and regulatory inspection, national regulatory policy, clinical trials, pharmacovigilance system, lot release, laboratory access and testing, market surveillance and controls, licensing premises. Yes, sir. They are having a checklist called a GBT checklist, global benchmarking tool consisting of 288 sub-indicators, 63 indicators, 9 major functions where you are going to have the assessment of regulators. हम भी आपको regulate करते हैं सर, हमको भी regulators है ऊपर से. So this is the thing. And right now it is only meant for vaccines, human vaccines. So WHO in near future they are planning to extend this NRI assessment to all medical products, including devices, diagnostics and regulatory authorities of different state government. You go to Chhattisgarh, उधर तो computer नहीं है. Go to Nagaland, internet ही काम नहीं करते. Go to Assam, है नहीं laboratory. So how we can get up with limited resources at the federal level under 5th year planning program? Government of India 1,782 crores granted in order to enhance the laboratories, in order to enhance the capacity building in the form of training. So things are changing. Now all 29 states, 9 union territories across the country covering 142 crores, 55 lakhs, 25,151 population. As I yesterday night, I don't know what is the Chinese population, but this thing population, but these are all the things, friends. So you can see this is the GSR and notification issued by government and scope, and these are all the important chapters. So you can go through this. How first chapter deals with the preliminary short titles and definitions, friends. then authorities and offenses, officers with respect to central licensing authority, delegation powers, etc. incorporated in chapter 2. Again, chapter 3, ethics committee. This is very, very important. Those who are doctors sitting in this hall. So institution ethics committee and independent ethics committee, that you can take care. Then chapter 5, clinical trial, BAB studies and of new drugs and investigation new drug friend. And chapter 6, compensation. Chapter 7, BAB study centers. Across the country, we are having more than 85 BAB centers. So we are having the guidance document. Those who want to go for the export of their molecules, class 2 and class 4 molecules, BCS class 2 and class 4 molecule. If you want to go for the regulatory endorsement, FDA approval means you have to carry out effective BAB studies with respect to proving equivalence of that product, tested products with the reference product. You have to prove your time maximum, area under constant C max, but they have to submit the data before their endorsement, product endorsement. And this is the chapter 8 with respect to the manufacturing of new drugs, investigation new drugs for clinical trial, and chapter 9 import of new drugs. So all this, देखो आप बाजू में उसका validity, and I have given the fees also, fees and import of manufacture of new drug for sale, for distribution, chapter 10, chapter 11, chapter 13. Yes, sir, you can go through that. And these are all the important schedules, 8 schedules. And these are all important forms. Which form you have to make application to FDA, that is CDSCO. Form CT41, CT01, CT02, 3, 4, 5. इतना अच्छा है देखो बाजू में आपको नहीं दिख रहे ना. So ये problem, no problem sir. So next time we will have one good symposium in IACT. रामजी साहब, ठीक है? And you can go through this sir. देखो पूरा है. I don't want to hammer you, okay? All these things. So preliminary clinical trial site, effectiveness and efficacy, ability to achieve desired effects already discussed, GCP formulated by CDSCO, adopted by Drugs Technically Advisory Board. APIs are very, very important, which are unapproved APIs. You have to take the testing, what we call the CT, clinical trial approval with respect to testing for analysis, examination, including BAB studies as per the NDCT rules. Unapproved APIs, you have to take unapproved formulation, unapproved API for the formulation development, unapproved formulation. So these are all important categories of the forms we have kept in our NDCT rules, sir, with the relevant fees. They have to make online application. We are going to approve as per the timeline. 7 days timeline, but we are issuing within 3 days. Andhra Pradesh and Telangana, during last 16 months, I have given 3,622 NOCs have given. 100% transparency, doctor. So NCEs. So here, NCE means what? Here, right, this thing, theme they have kept, not approved as a drug, proposed to be developed as a new drug for the first time by establishing its safety and efficacy. Is called as NCE. IND, investigation new drug, not approved for marketing as a drug in any country. That is IND. And orphan drug, a drug intended to treat a condition which affects not less than 5 lakh population in our country, is called as orphan drug. First time we have incorporated like US FDA in our NDCT rules, sir. Post-trial access. This is the important thing we have incorporated in NDCT rules. To a clinical trial participant, those who participated in that trial with the approval of ethics committee and sponsors should provide free of cost medication to those important participants if required as per the recommendation, sir. That is nothing but post-trial access. Then pharmacovigilance, everyone knows, Indian pharmacopoeia taking care, other drug reactions. We are also having science and technology deals with the adverse reaction, detection, correlation, archival, with respect to understanding principle, generation of the safety, passive surveillance, active surveillance is nothing but a pharmacovigilance, sir. A is already you know, adverse events, similar biological, similar means similar biological product is nothing but biological product similar in terms of quality, purity, safety, efficacy to an already approved reference biological product in ICS countries based on comparability is called as a similar biological product. Abhi, this is the definition one should understand. So don't get any confusion whenever you develop the novel formulation. This is the important definition of the new drug as per the rule 2W and rule 2S, investigation new drugs. New drug means a drug including active pharmaceutical ingredient or phytopharmaceutical drug. Earlier we were not having phytopharmaceutical drug, which has not been used in the country to any significant extent, except in accordance with provisions of the Act and rules thereunder, as per the conditions specified in the labeling thereof, has not been approved as a safe and efficacious by Central Licensing Authority with respect to its claim. It is nothing but a new drug. Number one. Phytopharmaceutical drug means what? Phytopharmaceutical drug means a drug of purified and standardized fraction assessed qualitatively, quantitatively with a defined minimum 4 bioactive or phytochemical compounds of an extract of a medicinal plant or its part for internal or external use in human beings or animal ailments for diagnosis, treatment, mitigation or prevention of any disease or disorder, but does not include drug administered through parenteral route. That is the meaning of phytopharmaceutical drug. Those who are engaged with the herbal research, plant medication, it is good for you, Doctor. 4 bioactive therapeutic mighties having intended use with respect to treating the particular disease or disorder. You take one plant extract having 100 mighties. We are not going to consider 100. Only out of 100, 4 important bioactive ingredients are important therapeutic mighties which are having more efficacy. Sir, at least minimum 4. दो भी है तो भी चलेगा. Depends upon if it is orphan drug, then definitely regulatory people will support, sir. At least minimum 4 bioactive compound of an extract of that medicinal plant, doctor. Then second one, drug approved by Central Licensing Authority for certain claims and proposed to be marketed with modified or new claims. A paracetamol मेरे पास है. I want to develop the liposoluble form. New drug. Simple. So this is the interpretation law, friends. And including indication. If you want to change indication, new drug. If you want to change route of administration from intramuscular to intravenous, new drug. If you want to change dosage from 500 to 1 grams, requires new drug. Dosage from oral to sublingual, new drug. So this is very, very clear, sir. Itna hone ke baad, madam, please interpret the law. This is the important definition for all young scientists like you, friends. Then third one is fixed dose combination. Fixed dose combination of 2 or more drugs, at least 2. Paracetamol with the zinc sulfate aayega. So another 10 minutes, sir. 10 minutes. Time hai na? 10 minutes, no? Okay, thank you, madam. So this is the important thing, so I want to. So here, fixed dose combination of 2 or more drugs approved separately for certain claim and proposed to be combined for the first time in the fixed ratio is nothing but a FDC which requires permission and we are considering as a new drug. Or where the ratio of ingredients is in approved combination is proposed to be changed with certain claims, then again new drug, including. Yes, sir. Sustained release, timed release, delayed release. Yes, sir. Fast acting release. Yes, sir. So that also considers a new drug, doctor. So including indication, route of administration, dosage form. So these are all important things as per the rule 2W definition of NDCT rules. Earlier it was under rule 122E, sir. Then fourth one is modified. I'm coming to your point, sir. Modified or sustained in form of drug or novel drug delivery system of any drug approved by Central Licensing Authority is also new drug. And fifth, all vaccines, recombinant deoxyribonucleic acid, RDA derived products, living modified organisms, monoclonal antibodies, stem cells products, including autologous allergic, what we call regenerative products, xenografts, that is gene therapeutic products, intended to be used as a drug, nothing but a new drug. And drugs other than referred to in subclasses 4 and 5, except 4, modified release and vaccine, shall continue to be new drugs for the period of 4 years from the date of their permission granted by the FDA. And the drugs referred to in subclasses 4 and 5 shall always remain as a new drug, sir. Okay, clear, sir? So as per the rule 2S, investigation new drug means a new chemical entity or biological entity or substance that has not been approved for marketing as a drug in any country called as investigation new drug. Unapproved drug is very clearly defined in rule 59. इतना volatile होते सर मैं पढ़ाई कर करके पागल हो गया. Still sometimes I forget. Nigrostriatal pathway में dopamine depletion होते, acetylcholine and serotonin down leads to all these complications. So we have to read so that you can understand all the principles and rules, sir. So ये orphan drug I already told, and we are not going to charge any fees for conducting clinical trial with respect to orphan drug. Provision for accelerated and expedited approval process, we are having 90 days timelines, and if it is for emergency use, within 30 days we are going to review the documents related to your scientific data submitted to CDSCO. This is the biomedical health research. Earlier there was no any regulation. Now there is a separate chapter, chapter 4, for regulation of BHR, which is dealt by DHR, Department of Health Research. Okay, friends, ये सब आपका है. And ethics committee for clinical trials, applicable rules, rule 6 to 14, constitution of the committee, registration validity renewal, application from CT01, CT02 for registration renewal, and timelines is 90 days with respect to review. Ethics committee means everyone knows. Ethics committee is a committee comprising of medical, scientific, non-medical, non-scientific members whose responsibility to ensure the protection of the right, safety, well-being of different subjects involved in the clinical trials, BAB studies, etc. And ये सब आपको हम hammer नहीं करना चाहते. I want to go with all important things. Academic clinical trial. बहुत लोग आते. Those who want to go for the publication, they can go for that. If you want to go for the marketing purpose, if you want to go for the regulatory permission, then you have to approach office of CDSCO. Academic clinical trial means clinical trial of a drug already approved for certain claim and initiated by any investigator, academic or research institution for new indication or new route of administration or new dose or dosage form, where the results of such a trial are intended to be used only for academic or research purpose or publication purpose and not for seeking approval of the licensing authority or regulatory agencies, is considered as a academic clinical trial. So these are all the things already completed, friends. Post-trial access, compensation policy we are having. We are all with you. Please go through the all my important slides, and I have given almost all important things with respect to advantages and all. Here, if you can see the important point, sorry. Kahan gaya? Can you come to last slide, please? So here, I would like to, because you know the new guidelines are very clear and we are not having any ambiguity, and with respect to that, and we are putting efforts, and credit goes to my boss. He is the true scientist come regulator, and I am happy for giving me the opportunity to share some thoughts with Ramji sir and Matsun Rao ji and our medical director, Dr. Kiran Martak ji. He is the now catalyst between ICMR, regulatory people, and scientists from Atal Incubation Center. So with this, I would like to conclude and congratulate all the scientists for your wonderful job in this important incubation center. We are all here to facilitate you. God bless you all for giving me the opportunity to share some of my little principles based on my 25 years of regulatory experience. Thank you, one and all, and thank you very much to our Sujitra madam also for widening the agenda. And I made a request. Now I have to go to office and I have to prepare few slides for the campaign 3, Swachhata Hai Seva. Because October 2nd is the Gandhi Jayanti, October 1st is the Sunday, and all my colleagues requested to come on first. Saturday, Sunday भी नहीं है, हम काम कर रहे हैं. And in case of private, things are going in different direction. So now government thought process is completely changing. That much I can say. Thank you very much, and we are all with you. Thank you, sir.
Thank you, Dr. Ramkishan, for a wonderful presentation on regulatory guidelines for drug discovery and development. Requesting you to stay back for 5 minutes. We'd like to now open our platform for any questions that our audience might have. Please feel free to ask any questions.
I'm sorry, I could not cover all my slides. Yes, sir.
Now, very quick question. Recently, one patient approached you, the organization you represent, and he gave you some feedback on the side effects. And based on that, you requested them, and then finally you asked them to take the entire batch of that product, Digene, out of the market. So how does it go about for scientists to understand?
So, sir, now we are having mobile app. Mobile app, Indian Pharmacopoeia Commission is the nodal agency with respect to reporting your adverse reactions, if any. And if you report to them, immediately you will get the one, number. And based on that, immediately we'll get the alert, notice to the CDSO, and CDSO people, DCJ sir will intimate to us, immediate go for the investigation, followed by recall within 24 hours if it is serious. Otherwise, we are having the recall procedure, separate standard operating procedure with respect to recall of product. And we'll carry out the investigation, we'll intimate to the company with respect to recalling all those products if it is in domestic and international also. We are having the similar mechanism for the fast track recalling of the products in case of the severity of the occurrence of those important molecules.
Once patient can report, there can be false.
How he can report to us? Because he is not aware of the CDSO. That's the problem, no, sir?
No, what I'm saying is, do you investigate? How do you do trials again for the product to find out that particular?
Trial is not required already, because established molecule already there is some adverse event, untoward reaction occurred with respect to that patient. We will carry out investigation whether any formulation defects or any factors influenced during the prescription practices, and what are the other drugs combined during their treatment, then severity of the disease, several factors involved in that. We are not doctors, but yeah. So we will have few experts involvement during our investigation, Dr. and we will have the submission. Based on that, regulatory action will be taken against to the respective manufacturer.
Thank you, sir, for the this good presentation and all. I'm not with a startup right now which is involved in a clinical trial or a drug or anything. I came to this symposium on a personal knowledge. I'm gaining personal knowledge. And frankly speaking, if I have to start something tomorrow with a new drug research or something with a startup, this is actually very prohibitive. If I'm already established with a Dr. Reddy's or something where they have a regulatory affairs department, I think they can cover most of the things what here. So now what I wanted to know is, for a startup, since we are in an incubator system, for a startup, what differentiation or what extra help? So see, we are founders, some of us, I'm not even a founder. So some of the founders I've seen, they go behind the investor, they get involved in the science, they get involved in the experiment research analysis. And this is actually seems to be an elephant task. I'm new to this field. I'm accepting that. But is there a way?
Madam, that is the reason why Veeda people are here, no? To connect. Veeda, CRO, Dr. Kiran Matriksa will tell. Now he is the right person. You understand from regulatory people having little limitation with respect to understanding this. But madam, darne ka nahi. You can do wonders. Now things are changing. Simple Google search में जो सब availability है. Only thing is we need to have some passion, and definitely with your scientific pool, you can extract information and you can take in a positive way, step by step. If you can go through, then knowledge,
Yes.
sky is the limit.
Yes.
There is any limitation.
So I think to the point, see, for the founders or wannabe founders for any startup, to become the companies like Reddy's or Shantas, or you need to raise funds, right? So basic understanding, not about the chapters in detail, but a larger landscape, a broad landscape, if you can understand, that will help you to raise grants, at least maybe some 100 to 500K or 300K. So I think that's where it may be an elephant today, but try to understand the tail of an elephant today to go through slowly through along with your startup journey. Thank you. Yeah.
Multiple committees, multiple organization. One side you're talking about ICMR, take permission from them. I mean, like there are ethic committees. There is then Serious Co and all. So can there be a single portal where everything can combine together?
Multiple committees means kuch nahi. If you want to get approval for IND, for example, investigation, IND committee is headed by ICMR DG.
Oh.
Regulatory is there. Based on the, because once the safety efficacy is proven, no madam, in your preclinical area, for example, biotech product, trastuzumab, for example, the RDNA product, once the preclinical safety efficacy established, then you have to approach the office of RCGM committee, falls under the DBT. So they will evaluate your data and they will guide you if it is okay. If it is not satisfactory, then again you have to repeat your data and you have to submit. Once the data is satisfactory, you can also have the parallel submission to CDSO, where you can have the permission for the clinical trial in case of ordinary. Just I am setting an example. So multiple committees से हम कम किया, पहले तो बहुत confusion था. That is the reason why Honorable Prime Minister of India is going to take care to have a Ministry of Pharmaceutical instead of approaching Ministry of Environment and Forest, Ministry of Commerce and Industry, Ministry of Science and Technology, Ministry of Health and Family Welfare, and Ministry of other important, 2-3 ministers, sir. Ministries. So we are putting efforts, but it will take some time. We cannot change the constitution of India, madam. So things are changing. Now we are having all dedicated portal, madam. You can upload the information and he'll get the requisite NOCs or approvals. That much I can say. Without any ambiguity, madam. How regulators are evaluating with respect to test license. Once the product is approved, you have to make application for the test license. Testing, analysis, examination, key quality attributes, critical quality attributes involved in that, whether product is going to be stable or not, based on physicochemical parameters, then method validation, then go for the pre-formulation, then formulation development, then analytical development, then goes for your commercial scale, first 3 batches, where you require perpetual licensing system in Form 25 or Form 28 with respect to manufacturing, sale, distribution in domestic or international commerce. Yeah, please, sir, please. Yes, sir.
So yeah, one question is like as a startup, so first like confusion what we'll get is like whether a clinical trial is required or not. Okay, because we don't know whether the regulatory pathway for that product. So even if we approach CDSE also, our experience is that actually the clarity sometimes is not emerging. Like does this
so now public relation officer and facilitation center also we are having, sir. In such type of case and in such type of molecule, we are having pre-submission and post-submission meetings at CDSO Delhi with relevant fees, sir. We are having the chapter 12 or 13. So you can approach, now you can have some inputs with respect to the clinical trials required or not required with respect to novel formulation developed at your startup company.
Okay, so can we follow through the Hyderabad office because we approach because almost 2-3 months.
Delhi office, sir.
Delhi office.
FDA Bhavan, Kotla Road, Kotla Road, near to Bhal Bhavan, New Delhi, 110011.
Okay.
India. And once you get the clinical trial, then when you want to go for the market authorization, you have to get then a regulatory inspection for the manufacturing facility inspection, GMP audit, what we call. Then field offices are involved.
So, for example, like for the digital health applications, like see, we are not like from somewhere as a pharma or medical, but as a software, as a medical device. So the regulations are absent. So, like, how do we go about it actually? Because we are doing academic clinical trials, but to get a marketing authorization,
clinical trial for research purpose or publication purpose, you will not be a person. If it is for the regulatory approval, then only you have to approach. If it is for the clinical trial, academic this company, you have to carry, go for the publication, give the impact factor 6.5, okay, Elsevier journal. But if it is marketing purpose, authorization required anything, then you have to approach CDSU Central Listing Authority, sir.
Thank you.
Thank you. Thank you, Dr. Ramkishan. I'd like to invite Dr. Kiran Mathur, Director of Medical and Regulatory Affairs at Veeda Clinical Research Limited, to present a memento to Dr. Ramkishan.
This is dedicated to all of us. This is also violation as per the FDA, rule number 6 of WHO. So I can keep here. So ask me, irrigation making easy. But we are also having vital organs, we are also having family, kids. Thank you, Andhra. So excellent brains, excellent scientific food, and leadership factor engagement global destination for all life sciences. By 2030, they are going to reach 100 billion, and 2047, 300 billion they are going to come. Out of 1,237 billion dollars, only 2 products. Excellent scientists like you can take me to serve vulnerable population in order to promote intervention. Thank you.
Thank you. Thank you, Dr. Mathur, and thank you, Dr. Ramkishan. We will now be heading to our next speaker, and I am extremely delighted to extend an invitation to Dr. Jagannath Kota for his talk on biomarkers in clinical trials. Dr. Jagannath Kota is currently designated as the site head at Novartis Institutes for Biomedical Research in India, overseeing a team of 22 scientists with diverse expertise in clinical pharmacology, toxicology, modeling and simulation, and analytical sciences. With a strong background in drug discovery and development, he has steered multiple projects from conception to market success. Dr. Kota is a key figure in Novartis' external collaboration efforts, fostering talent within and outside the organization. He holds a PhD in pharmaceutical sciences from Monash University, Australia. Thank you, Dr. Ramkishan.
Thank you, sir. All the best.
I would now like to invite Dr. Ramji to present an honor to Dr. Jagannath. Can we have a round of applause for him? Yes, you may begin your presentation. Thank you, Dr. Ramji.
Show to you. This one, okay. Just advance. Yeah, just this one. Goes to the next one. Okay. Thank you. All right. Very good afternoon. I am, first of all, my apologies. I am a little crunch on time. I had a hard stop at 4. I requested people who are my backup to see if they can manage. But if I get a buzz and if I am required to be there, I have to take apologies and I may have to step out. But I will make sure that I cover the concepts as much as possible. Okay. First of all, warm welcome and thanks to the incredible minds here for accepting to this knowledge sharing session. I'll try to share as much as I know, as much as I learn. And then after a short break of a couple of calls that I may have, I'll be open to any further interaction. Okay. So whatever I talk today is my own personal opinion from literature articles and publications that I have read through, and not anything related to either Novartis processes or Novartis way of working. I'm here as Jagannath and not as an employee of Novartis. Okay, thank you. Now, biomarkers in general, there are many biomarkers that are right now available in the market, but are all those biomarkers validated? And are all those biomarkers really useful in getting a tangible outcome of the clinical trials that we conduct? The answer may be a no, but there are certain biomarkers that really give us a concluding evidence on our way forward, which is one example I'm going to talk through today in this session. So biomarkers principally, if I summarize this slide in one quick snapshot, they enable us in taking the right decision, guided through data, guided through information that is available through multiple analytical techniques that we apply. And these biomarkers either help us in fine-tuning our clinical trials or interpreting and understanding the results of our clinical trial. Either way, it is pre or post-study, they enable a scientific understanding of what the data are telling us. Okay, so there are no data that are useless. All data that we generate are somehow useful. But it is up to us as a scientific community to see how best we can utilize the data that are generated. Okay, human biosamples are a treasure, and that's the primary starting point for us to start research, particularly in the area of biomarkers. Okay. Now, there are many phases. I hope these slides are better visible than the other one. But if they are not, my apologies for that again. I'll try to see if I can, take you through. But basically, the concept of this slide is to show how we come out with the mechanism. Okay, we have different mechanisms, different pathways that we research all through our career. But in the pathway, in the principal pathway of a particular disease or the disease manifestation, what is the point in that pathway? Okay, what is the specific point in that pathway which is of our interest to enable building a biomarker strategy is what we need to decide. This can be at the stage of drug discovery. This can be at the stage of where we are in midway of the clinical development. It can be any stage. There are instances where we serendipitously discover that there is a biomarker which is significantly altered in terms of the way it manifests in midway of clinical trials. So attention to detail and trying to understand what all the data are telling us is extremely important. Okay, so that's the principle of this slide. Primarily, it's about understanding the mechanism, understanding the upstream and downstream effects of a pathway or where we impact the pathway, and then subsequently interrelating or correlating to different mechanisms. So in the example that I give you, you will be able to appreciate the fact on how we need to, correlate two things that are a little different or parallel, but some way connected to enable each other. Okay, I will leave you with thoughts here, but these thoughts will get clarified as we move or advance through the presentation. Now, here is one example, BRAF V600E inhibition, particularly in melanoma. So now if you see the plot here on the left side, you have 80% response in melanoma, but you only see 5.2% in colorectal cancer. And this difference in the effectiveness by inhibition of BRAF V600 is principally because of the differentiation in pathways and differentiation on the feedback changes that happen where there is upregulation of some pathway because of the inhibition of BRAF V600. So that I will talk to you subsequently in the next slides. But what is very important here is we don't stop. We don't stop after this result and say, okay, this inhibition is not likely to work in colorectal cancer. Okay, if as scientists, if we stop there, research does not progress. That's the key message on this slide. Now, if you see this slide, you see different cell lines that were tested for colorectal cancer, and down below, A375 is the melanoma effect. Now, if you see BRAF inhibition has a clear, distinct difference in the way you see the outcome in melanoma versus colorectal cancer. So difference in lines that express, VSCV432 is a line that significantly expresses a system which is called epithelial cell receptor. This expression of this receptor, EGRF, is the reason why you do not see an outcome in colorectal cancer versus melanoma. It's because of the build. It's because of what is the primary route for generation of the cells. So as we all know, there are different progenitors for different cellular systems in our body. So that's the reason why you see a very clear difference in the way the disease manifests, or the way the pathway is different across both. Okay, now here we are talking about vemurafenib, which is the drug of interest, which was tested for inhibition of BRAF V600. The mutant BRAF V600 and the mutation that is a feedback mechanism that happens in colorectal cancer is contributing to low response. Yes. Yes. Correct. Yes. Thank you. So if you see here, this is a very clear representation of what we have seen in the previous graph. You see the first three panels of the dishes, cell culture media. On the top three rows, you see that there is no inhibition. But in the bottom row, which is A375, which is melanoma cells, you see a very clear effect of vemurafenib. Okay, that's because that's exactly related to the mechanism that I'll be talking about now. So here's the mechanism. Now, typically, the way it happens is when you have, let me try to pull the pointer. All right, I hope this is making it a little better. Now, what happens, typically is when we see BRAF inhibition, and that particularly acts at the RAF pathway. And that phosphorylation subsequently affecting the MEK and ERK pathway results in positive effect in melanoma. What happens in colorectal cancer is because of the, TGF alpha, okay, TGF alpha in fact acts and then upregulates EGFR. The upregulation of EGFR results in RAS phosphorylation. Thereby, this is facilitated by generation of phosphorylation of RAS and then phosphorylation of RAF. That is the reason why we do not see efficacy of vemurafenib in colorectal cancer. Okay, so that's how this mechanism of resistance that is generated upon treatment of colorectal cancer with vemurafenib because of EGFR is resulting in no effect in colorectal cancer. Yeah. Yeah, yeah. Right. Correct. Right. Yes. Yeah. Correct. Yes. I fully agree. Yes, sure. Thank you. Okay, so we know there is a difference. We know there is a gap in terms of the mechanism, and we know there is a mechanism that is resulting in the resistance or probably no effect in colorectal cancer. How do we explore that? So, in order to explore, we try to culture two different, media. In one media, we had Atal Genome for shRNA screening, and in the other media, we had phosphate shRNA screening. The purpose of these two was different. The purpose of genome assay and purpose of phosphate assay are two different mechanisms. I'll talk about it in when I click next. But the reason why we are doing this assay is to interlink and try to understand what is our interest and what are the pathways that we need to be focusing on to see positive effect in colorectal cancer, because the principal mechanism should be useful. There is a secondary pathway that requires to be inhibited for us to see that remorafenib works in colorectal cancer. What is it? That is what we would like to explore. Now, when we culture by infecting the cell bases with shRNA genome library and shRNA phosphate library, there was a clear difference between control and remorafenib, both in the genome as well as phosphatase groups. Now, what is the difference? The difference we have seen, if I can take the phosphatase group, there were altogether 290 plus assessments that happened. In the 298 plus assessments, thank you, there were only 3 that showed an inhibition of a desirable extent, and that is the SHP2 pathway. And only 2 in the SHP2 pathway and 3 in the EGFR pathway have shown a positive inhibition. What that essentially means is these pathways can be considered for us to really understand and demarcate where we need to explore subsequently. Okay. All right. So now we know there are 2 different pathways that we could consider. Okay, which is BRAF, EGFR, and SLP2. Now, within the scope of these 2, if you see EGFR and SHP2 inhibition, which confers sensitivity to BRAF, if inhibited simultaneously, if you see the plots here, okay, that's the control group, which is the solid squares, and cetuximab, which is the EGFR inhibitor, you see that as the yellow line. PLX is the remorafenib, and you see that as the pink line. But when you combine BRAF inhibition and EGFR inhibition, you see a clear difference in the way the manifestation is observed in the patient population. Okay. Similarly, if you observe knockout, which is the SHP2 knockout. So this is the control vehicle for SHP2 knockout. You see that blue or green, whichever color you see here. And then you see SHP2 knockout, which when treated with remorafenib. You see a difference which is similar to what you have observed here. Now, what does this tell us? This tells us that in both SHP2 as well as EGFR, there is a clear difference when these combinations are treated and in combination with remorafenib, where there is a positive outcome in terms of tumor suppression. Okay. This is a little more detail into what I talked about in the previous slide. These blocks are not very clear for people sitting. I'm really sorry about that. But the purpose of this slide is very simple. So we have gemcitabine, we have cetuximab, and then we have PLX4032, which is the treatment that I've been talking about. Now, when you see this control group and compare the control group with PLX4032 for all the pathways, versus when you see PLX4032 in combination with either cetuximab or gemcitabine, there is a clear difference in the outcome in the Western blots. Okay, that again speaks into what we have seen in the previous slides and tells us that SHP2 and the reactivity associated with SHP2 can serve as a very strong and positive biomarker for us to correlate observations thereafter. It doesn't necessarily limit to one of the indication, but it can extend to any of those kinase pathways across the board, because majority kinase pathways are associated with SHP2. So it principally could apply to all kinase pathways if considered wherever appropriate. Okay. Now, similar is the case, just like this, we have demonstrated the difference with the wild type as well as the knockout. So we have two different knockout categories, which is B9 and B19. In cases with respect to SHP2 pathway, there was a clear differentiation on the positive effect of the combinations. Now, all we have talked about till now is represented here very clearly in this pathway. Now, when you have the ERK facilitating, SHP2 and thereby conversion of RAS to phosphorylation by guanosine biphosphate to guanosine triphosphate, and then phosphorylation of MEK ERK, thereby the EGF and EGFR and SHP2 together are contributing to loss of effect. Now, if you look at the SHP2 in RTK activation, it's very clear, when SHP2 initially is closed form, which is inactive, and then, when the growth factor contributes to activation of SHP2, the opening of SHP2 and by phosphorylation results in RAS activation, and that again contributes to the effect that we have seen in the previous slides. Now, when we knock out both, when we do not have SHP2 by knockout or probably or inhibition or loss of SHP2 and then drug inhibition, we see that there is no RAS activation, and we clearly see that the PLX4032 is active and produces its effect by inhibition of RAF. Okay, it sounds very simple, but the research that went into building this hypothesis and coming to this conclusion that these pathways could potentially complement with each other and then result in an outcome, which I show you in the next subsequent slides. So we have seen till now that EGFR levels determine response to BRAF inhibition. And then we have also seen that SHP2 inhibition also subsequently shows positive response to BRAF inhibition. Now, this slide, what we tried to include different melanoma categories. So melanoma, colorectal cancer, and thyroid carcinoma. In all these categories, the beauty of this work is there were couple of colorectal cancer categories like COLO741 and OHCO1, where there was an inhibition seen in colorectal cancer, which is a partial observation. It's not in all patient population, but there are obviously exceptions to each patient categories or each patient population, right? This is because of the partial or partially lower expression of EGFR in these patients, which resulted in a positive outcome when treated with remdesivir. Okay, these are the exceptions that we need to really bear in mind in the way in the way we interpret the data. These should not be interpreted in a way that there is a positive outcome in colorectal cancer, but the way that we need to interpret is there are cell types or cell subtypes where the expression of EGFR is not optimal, and you could see a positive outcome in colorectal cancer. Okay. Now, I really apologize. This is such a pathetic slide because it just cannot see anything. I'll tell you what it means. What we intended to convey here is we have a 6-patient population. Okay, if it is possible for you to see the treatment with remdesivir because of EGFR activation, it has a brown stain. When you see the slides, when the slides are shared with you, you can see this slide number. It should be slide number 11. You see brown staining. Okay, brown staining shows that there is EGFR activation, and thereby you see no effect of remdesivir unless you give the combination of EGFR inhibition. That's the purpose of this slide. So BRAF melanoma also sometimes, not just colorectal cancer, but even in melanoma, there have been patients who had an expression of EGFR because of which we have seen 6 out of 16 patient samples had EGFR expression post vemurafenib treatment, and these patients correspond to low response when treated alone with vemurafenib. Although in melanoma, we have seen 81% success in treatment of melanoma, okay, with vemurafenib. Okay, that's the difference. So like what we have seen in terms of EGFR expression, which is lower in colorectal cancer, there are instances where melanoma patients, because of some etiology, have expression of EGFR, and there you see loss of, low efficacy of treatments like vemurafenib for BRAF inhibition. Okay. Now, again, the same thing. Like SHP2, like I said, SHP2 is a great biomarker. It has very broad applicability, and there are many instances where we could continue to use this as a strong biomarker. But there are these slides which talk about, the parental cells which acquire resistance to vemurafenib, and this brown staining, and excess staining also clearly indicates that there is excessive expression of SHP2. On the contrary, if you see the knockout cells, there is slower expression of SHP2, which again ties back to the hypothesis that we are trying to understand here. Okay. Now, since this slide is not very clear, the message is very important. SHP2, immunohistochemistry staining is really helpful in identifying the RTK-dependent resistance mechanism. And that's the principal mechanism we have applied to really understand where we need to be paying more attention to. Now, these are the publications, and I'll be happy to share the publications that these slides are coming from. These are very interesting publications. They very, very systematically talk about every step that was a part of the research that was done. Every figure has been very clearly detailed. There is nothing that is not clear from these publications and presentations. I will share all the 3 or 4 publications with the audience team. Now, we didn't stop there. The outcome of this research in terms of the applicability of inhibition was translated to at least 3 different clinical trials by Array Pharma, GSK, and Roche. They used combinations of BRAF inhibition and EGFR inhibition or SHP2 inhibition in their clinical trials, and there is this Array Pharma phase 3 trial that has really come out with very strong data that I'll present in the subsequent slide, which talks into the value that has been generated by this research. Now, that's the slide I was talking about by Array Pharma. Then treatment with encorafenib, binimetinib, and cetuximab for BRAF inhibition was tested. So if you see the 2 combination, and the control group was folinic acid, fluorouracil, and irinotecan. If you see the control group, there is a clear lower response versus the 3 combination and 4 combination, which again speaks into these data. Now, if you want to focus on the number of risks, oncology as an indication, the number of risks would obviously be not different between control and treatment group. It's about the quality of life, or probably partial prolongation of life, that we principally focus on, and which is also evident here. With the triple combination, the extension of life in months was 8 to 11 months versus the control group, which was 4.8 to 6.6 months. And then if you see the dual treatment group, it was 8.4 months versus 5.4 months. The difference is in months, but every month for an oncology patient counts in terms of life expectancy or prolonging life. And that's the principle we have to really apply here. So this is clearly success on treatment. So if I have to summarize, I would say that, mutation, tumors like melanoma, colorectal cancer, or thyroid cancer, thyroid predominantly manifests as 50% effect or 50% no effect. When we look at BRAF inhibition, they do not respond uniformly. As we have seen, there are CRC patients who respond, there are melanoma patients who do not respond. It's a hybrid kind of treatment option. And then functional genetic screening, of course, what you have seen in the slides through different genetic screening of the part of the hairpin that was utilized, part of the hairpin of the gene that was utilized to see what are our primary markers that we need to be focusing on. Like you heard, we focused on more than 250 of them, and only 3 of them came out to be useful for our interpretation of data. So principally identified EGFR as a biomarker pathway to partner with BRAF is an important observation. And to conclude, SHP2 is a very important biomarker which does not restrict to a specific area, but it can be broadly applicable across all kinases. With that, I conclude. Thank you.
Thank you, Dr. Gupta, for a wonderful presentation. I would now like to open our platform for Q&As. Would request the audience to ask any questions that they might have.
MicroRNAs being used for disease progression in cancer, as well as for your drug efficacy, right? They are being used widely for, particularly in oncology cancer, where which drug is working has more efficacy, which has less efficacy based on the microRNA in the blood. That's first part of the question. Second part is, what are your thoughts about the new, new approach, or approach is already there, but in UK they are using now for, alerting your immune system through MABs, and they are using it. If you've been in the news, you've read last 2 months. So those 2 things, if you can clarify. Thank you.
Sure. Within the limited understanding I have, the targeting strategy works well when we use microorganisms to a great extent. It helps, but I wouldn't have tremendous knowledge there to be able to share some insights in there. But pardon me, what was your second question? Yeah. Right. New therapies. Right. Monoclonal antibodies. Yeah. So, see, practically speaking, monoclonal antibodies, as we see, they are kind of new. They have been well established. They are more targeted, more focused. But more so the way we are advancing into radioligand therapy, we are developing therapies which are more targeted to reach the cancer site through a radioligand. There is a lot of research happening. There is tremendous work happening in gene therapy, cell-based assays, B-cell therapies that are working very well. So I would say monoclonal antibodies are probably, the known way or approaches, but as innovators, as the future of medicine, as people who would be very much interested into research, I would rather focus more on targeting therapies for cancers. Thank you.
Any other questions?
Yeah.
On CAR cells, CAR therapy, CAR T cells.
Yep.
And I think Novartis is involved in the research and development of this. So if you can help us in understanding that.
Sure. Novartis has been significantly investing in CAR T therapy, likewise are many other companies. There is strong promise, but it's the more important part is in countries like India, how we can apply an optimal strategy to make these kind of treatments affordable. That would be more important. Thank you.
Any last questions? There's one more question.
So I would like to ask, in phase 1 trials, how simulation can help to understand the disease progress and everything in the human, like in the patients, and how we are working on the simulation part.
Sure. I can add, and then I'll probably ask my colleague Jayant to also add a little bit on the simulation perspective. Simulation has been, in fact, playing a very good role. I wouldn't claim a lot on phase 1. It's playing more on the phase 2B, which is dose range finding. But there are examples that we have witnessed where there is an optimization of dose strategy, getting the dose right. There we have applied simulation to really understand how the biopharmaceutics, which is the physicochemical properties of the compound, complement with what we anticipate to expect in patients or healthy subjects when we initiate the phase 1 trials. These correlations have really helped in reducing the dose that we would be projecting as the first in human trial. So the simulation has certainly helped. Do you want to add anything, Jayant?
So it starts from the discovery phase also, right? Whenever we get some kind of data from, suppose there are data from pharmacodynamics, there are data on pharmacokinetics, and there we combine the data from formulation, right? Whatever the solubility and permeability information that we get. So based on that, we try to predict the dose at each stage, right? We have some early information about the solubility and permeability. We get the data, we simulate it, and we understand where we want to go. If that leads to a kind of formulation development aspects, that suppose the solubility will not work for us. Then we go to our formulation expert, we ask them to modify the formulation. So, and once we modify the formulation, we reach that state, then we try, okay, or if everything works, then can we use this for FIH, right? First in human study. So every stage we do modeling and simulation to reach to next step. Yeah, it includes from early discovery, late discovery, early development, and to late development also. So every stage we do modeling and simulation exercise.
There are many examples. Correct. There have been many examples, actually. There have been many examples where bioburden has been reduced, not just in the clinic, but also in non-clinical. There was one study, I cannot define more, but the number of rabbits required in a particular study was 300 plus. The number of rabbits utilized was 25, based on modeling and simulation.
Thank you. Thank you, Dr. Jagannath. I would like to call upon Dr. Mathak to present a token of appreciation to Dr. Jagannath. Thank you, Dr. Mathak. Thank you, Dr. Jagannath. Moving on, our next speaker, I have the honor of inviting Dr. Suchita Makhan to deliver a focused talk on ICMR initiatives for strengthening the medtech ecosystem in way forward. Dr. Suchita Makhan, scientist and mission in charge of India's Medical Device and Diagnostics Mission Secretariat under the Indian Council of Medical Research, possesses over 15 years of expertise in public policy, intellectual property, and technology transfer, with a specialization in medical devices and diagnostics. She has been instrumental in crafting policies to promote innovation and entrepreneurship in public health. Dr. Makhan is acclaimed as a thought leader representing India's healthcare interests on global stages. Her contributions led to her selection as an emerging leader for biomedical innovation and entrepreneurship via US Department of State's IVLP. She holds a doctorate and is a registered technology transfer professional, recognized internationally for her dedication to technology transfer and substantial publication record. I'd like to invite Dr. Suchita on stage. Along with it, I'd like to invite Mr. Rajkumar Agrawal to honor and acknowledge Dr. Suchita. Over to you, Dr. Suchita.
This is for pointing. Fine. Thank you, CCMD and Veeda for inviting me here. And it's really great to collaborate with other science agencies, CSR, ICMR, DST. Now, all of us are working together for fostering Make in India in all the sectors. So I'll apologize. Primarily, I think some of the slides may not be visible, and I have included a lot of animations as well. So maybe you will have to bear with me. So basically, I thought that these are not there. नहीं आ रहा। You all will maybe need to change some gears because we have been talking a lot about the new chemical, new NCEs, the vaccines, the therapeutics. What I'm going to talk about is medical devices and diagnostics, which are the prime things. The potential for this sector has been recognized by the Government of India, and it has been included as a sunrise sector under the Make in India initiative of the Government of India. This sector has huge potential, and at the same time, we all know that it is about 80% import dependent sector. These we can call as the low hanging fruits, which we can actually innovate and deliver to the market, and they can actually impact the public at large, which we have seen very clearly in COVID times, wherein we didn't have any diagnostic capacity to make COVID kits, but immediately we could make huge lot of kits across the country. So that capability lies within us. Interestingly, India, we all know, is 4th largest in Asia in terms of medical devices and diagnostics. The sector is currently valued at $12 billion, but we all know that it is poised to be a $50 billion sector by 2030. An important aspect I wanted to bring across you all was that the growth rate which we are seeing at today in medical device sector, we see that India is growing at a CAGR of 12% in medtech sector, while the world is seeing 7% CAGR growth. So we are well ahead. The sector is huge potential and it's growing exponentially. So that's a very important aspect I thought I'll share with you all, that we are quite ahead when it comes to medtech sector growth. As we compare with even the largest economies of the world like US, Europe, we have much more greater growth rate in the medtech sector. But why are we not seeing a lot of products in the market? Particularly, I'm talking about the government funding, primarily to government funding support. We thought that we all working in the medtech sector would appreciate that this is a typical pathway that we follow. When it comes to medtech innovation, it always starts with some kind of funding. Then we start with 3I process of by design, that we try to look at the problem that we want to solve. We identify the unmet need, and then we try to invent around it, develop proof of concepts, and then we try to implement it by scaling it up and then trying it across and commercializing it. So these are typically the steps of medtech innovation pathway, from funding, identify, invent, and implement. But what we have seen typically is that lot of technologies are getting developed. We get funding support for some idea. It reaches the proof of concept stage, and then what happens? Since we do not have clear hand-holding support for taking technologies to the next level, and maybe we get another round of funding. So typically we see a group of proof of concepts bucketed together, and we don't see technologies going to the next phase of invent and implement, because there are very great valleys of depth in the medtech innovation pathway, which we really need to cross in a collaborative manner. So keeping that in mind, ICMR did a kind of survey and talked to a lot of medtech entrepreneurs and tried to talk to other ministries as well on what can ICMR do to bridge this gap. So that's how the concept of Medical Device and Diagnostics Mission Secretariat, which I'm heading at ICMR headquarters, came up, wherein we thought that ICMR, being the leading biomedical research organization of the country, can we join hands with innovators, maybe leading institutes across the country who are developing day in and day out, innovating day in and day out, and help them with some kind of regulatory strategy, hand-holding, clinical studies, validation, and maybe if we give hand-holding support to 100, maybe 10, 20 can cross the valleys of death. And ICMR also has a unique strength of doing health technology assessment to effectively evaluate the cost effectiveness of those products as far as public health procurement is concerned. And then can we recommend some of those products which are clearly aligned with the national health programs for absorption in the program, which will be a very good success. And I think that if we are able to do so, but we have that potential, we have been doing this in bits and pieces for various diagnostics. For example, MolBio, we have recommended their products for TB diagnosis are being procured in volumes. CTB is a product by Serum Institute, which we did validation and operational feasibility is in volumes being purchased by the government. So we have that inherent strength, but we are doing it bits and pieces. With MDMS on board, we will be doing it in an upscale manner. And that's how the ICMR headquarters is committed. Yeah. I have a separate slide on that. Health technology assessment is a globally accepted model. It is done internationally also. It's a very systematic approach to evaluate the cost effectiveness and appropriateness of the product for public procurement. So Department of Health Research, under which ICMR lies, this is our host department. This does health technology assessment, and you can just see many reports which they have done. That too, we do everything through collaboration. I'll share more details about HT in my subsequent slides. So we all know that in medtech sector, it's just not one kind of support won't help. So we are trying to have a multi-pronged approach to give hand-holding support to this sector. This is the mandate of MDMS. This is the vision and mandate, and the yellow portion here, particularly TRL 4 to 7, is what we are targeting, because we know that institutes have huge lot of strengths. CCMB, all institutes have great strengths in doing the R&D. We have no doubt about it. And we are just an extension of, as government, if we can enable some of those technologies, that will be a great task we can do. So we are particularly focusing among the different verticals we are trying to address. The preclinical studies, policies, we also need to have the right appropriate policies and guidelines in place in which ICMR has a mastery. So those on guidelines also we are working on policy facilitation, we are working, and on the preclinical clinical part, and HTA is where we are primarily focusing on. Another interesting aspect which we saw is that for medtech innovation particularly, we have to have interdisciplinary research. Medical institutes working alone and engineering institutes working along with the CROs or that startup ecosystem, all of them need to work together. So we are also trying to focus on supporting proposals which foster interdisciplinary research. So in that aspect, we have set up various centers of excellence across the country. 7 to 8 centers we have set up as of now, which are typically based on interdisciplinary research. Medical doctors and engineers have come together. In fact, when we launched this scheme, we said that we are setting up ICMR DHR COEs at IITs. So ICMR DHR is itself going to IITs so as to facilitate and give them guidance on what is it that is the need of the hour for public health. So this is an aspect. In last 2 years we have created these centers, and we are very open to strengthen this kind of an ecosystem across the country. Not disease focused, actually we work with them very closely. We go to these IITs and try to identify the technologies which have alignment with NHM. So whatever we got from them, every year we go and try to find out more technologies which have potential alignment. So the technologies which have more relevance in PHCs, CHCs, which can have volumes of people to be impacted, are our priority rather than the technologies which are more aligned, important for tertiary health. Because we know that 70% of Indian population is rural health. So this is a strategic collaboration because we had to start somewhere. So this is an experiment that we are doing. We did with IITs, and they have technical strengths, and we are trying to give them hand-holding support for validation and mentoring in terms of regulatory facilitation. I'll just give you a few examples of some technologies which are coming out from the system to give you an idea on the kind of technologies we are looking at. In fact, I also have some case studies to support the way we try to help the entrepreneurs. So we can, the second one, we can look at this. This is a ophthalmic screening device. The second one, we know that slit lamp is the main instrument which is used for ophthalmic screening, but it is very much skill intensive. So one of the startups at IIT Madras developed this ophthalmic screening device, which is smartphone based device, and you can just carry it along with you in a suitcase to any place. It's very cost effective. And this is, we have compared it with the slit lamp. It works as good as slit lamp, and it's very cost effective and very easy to carry. So these kind of technologies, and the technician will take a picture, and ultimately we have it will be backed by a doctors sitting at the tertiary healthcare or maybe at a district hospital, giving the recommendation on if it has to be taken to the next stage. So it is basically a screening tool to help prevent the preventable blindness in the country. So it is aligned with the National Program of Control of Blindness and National Program for primarily with blindness. Other product is of IIT Hyderabad product. Yeah, both cataract. It can look at both the cataract as well as the corneal defects also it can see. And we portrayed this product in G20 also. It gained a lot of traction. Other technologies, just an example is NemoCare. I think it's an Hyderabad technology, so you guys might be knowing it's from IIT Hyderabad. We have a Center of Excellence at IIT Hyderabad also. So this is good technology for maternal and child health, which we are supporting. We knew that when it comes to large animal studies, particularly for medical devices, the infrastructure was limited. So we have set up a facility at AMTZ specifically for doing large animal studies for medical devices. So this is operational. You can very well use this facility. And since we are in Hyderabad, I am sure you might be aware about the NARF, ICMR NARF, which is the largest facility. It is 100-acre plus facility, in fact best in Asia facility, which is there in IIT Hyderabad, in Hyderabad itself. And this facility is capable of doing all kind of preclinical studies, from small animal to large animal, any kind of study. So this has been launched, and yeah, very well for preclinical studies, animal studies, not clinical. Preclinical. Basically, in this presentation, I'll tell you that we have different kind of networks for facilitating preclinical studies, clinical studies, health technology assessment, and some centers we have created for technology development. Other centers we are creating for doing the validation, and we do open call. When we select those technologies, we have open call wherein people can join us. The institutes can join us if they want to develop center of excellence for doing preclinical, clinical study. And ICMR will be there to help develop the right protocols. What is required by the health, by the government. We will have a technical committee sitting with the scientists, with the innovators, to refine the clinical study protocol which is required, and guide you with the regulatory strategy, and connect you with the right centers which are on board with ICMR to take you to the next level, preclinical, clinical studies. HTA. Yeah, CDC also, in fact, interestingly, is on board with us. So we help do the regulatory strategy beforehand. So that's the kind of thing we are trying to facilitate as government. In fact, you won't have to pay anything. ICMR will be supporting you completely end to end. But for that, you need to write a proposal. And once it's approved, then entire funding will be through ICMR system. Sure. We have actually my division is looking at devices and diagnostics, but we have a clinical trial unit separately set up within the ICMR system. So we do work for vaccines and therapeutics also, but devices, we were not into devices earlier. So this is what I'm trying to share, that we are trying to now have a right ecosystem built, utilizing the strengths of ICMR to help entrepreneurs. MDMS, the division I'm talking about, is looking only at this, but we have other divisions who are looking at vaccines and therapeutics also. In the Hyderabad, it is, yes, Hyderabad Center will take care of animal studies of any kind of biological entity. It's not only devices. So this is a clinical trial network we have set up across the country, having 50 odd sites. This information is available on our website, and this presentation also I'll share with Ramji. He can share with you all. So we use this network of our clinical trial sites to navigate through the clinical studies. This is a very new initiative regarding phase 1 clinical trial support. This was not there earlier. We had a lot of deliberations, interministerial also, we had a lot of deliberations that how do new technologies come into the market. I mean, they should go to the next level, and this is a very risky proposition. So if the innovators go straight away to a hospital, nobody is interested to do phase 1. So this is what we have just initiated. We have set up a phase 1 clinical trial support unit within the ICMR system. And recently we did the first expression of interest, wherein from all India level we had requested for proposals, particularly for devices, vaccines, therapeutic molecules, for which they need support from ICMR for phase 1 studies. And ICMR has set up 3-4 centers across the country with whom we are working very closely to develop the SOPs, the right way they have to do it, and we'll be strengthening them with the whatever the capacity they need, so as to do these studies, phase 1 studies. So that EOI is done, first call has been closed, and we have few proposals coming up. But I'm just telling you that this phase 1 call for proposals will also keep coming, at least once in a year it will come. So if we have something very new for devices, it will be class C and D, because A and B won't need any human clinical studies. Particularly C and D are the ones which are risky devices in which we feel we can assist. This is the health technology assessment I was talking about. This also we do. We have a lot of centers across the country, and you can go through nice guidelines. I mean, internationally, HTA is recognized as a standard methodology for assessing appropriateness and cost effectiveness of new interventions for public procurement. So this HTA studies also we do, but as of now we are doing it for only those technologies which are recommended to us by the government. So somewhere we had discussions with the startups, and they are also interested. So I recommended that maybe if it can get routed through the government, then the topic we are mandated to adopt for doing these studies. But we will be scaling it up. This HTA evaluation also will be scaling it up maybe in near future. So if we can summarize, we have centers set up across the country for technology development under MDMS. We have testing facility also in IIT Delhi. We are setting up a national facility for particularly for pilot batch manufacturing of products which can in CGMP compliant in clean rooms, which can go for clinical studies. That was again a very big gap in the medtech innovation pathway. So this national facility we have already supported at IIT Delhi. It is under process. Then preclinical research facilities we are trying to put in and as I mentioned, we have a clinical trial network. We have a network for doing health technology assessment. So when it comes to the last mile TRL4 and above, ICMR does have the necessary capabilities along with our own institutes and the collaborators, and we'll be very happy to collaborate. If I talk to Ramji, if we have some good promising technologies coming from the CSIR system, they can come together in the form of interdisciplinary research. We'll be very happy to support. So it is not only the medtech product development which we are trying to foster, as the regulator said in the earlier talks. Creating enabling policies and guidelines is a very, very important thing in which ICMR has been venturing for, I would say, since the day we were conceptualized. So depending upon the need of the country, we come up with new and new guidelines. So for example, in my subsequent slide, I'll tell you that we all know that when we go to doctors, if some startup company or some industry goes to medical college, they will straight away refuse that will not touch your product, because we are not mandated to work with the industry, and we have to maintain an arm's length. operations. So ICMR, our earlier DG understood that this is a clear bottleneck, and therefore we came out with ICMR DHR policy on biomedical entrepreneurship, particularly for medical doctors. So now that policy has been launched by the Honorable Minister, and by virtue of which, like engineering colleges, like IITs, now doctors can also form startup companies. They can take sabbatical. Medical institutes can take share in the equity. It's a very forward-looking policy. And we have recommended that every institute, every medical college should look at this policy because it's a national policy and make their own policy, startup policy, so that it is more implementable in their institute. So AIMS is working hard. We are helping them develop their own startup policy. And we are just waiting for that to release, because then that document can act as a standard document for others to adopt. So we are working closely with AIMS. AIMS Jodhpur is also at a very advanced stage of developing their own startup policy based on the entrepreneurship policy released by the minister. So these are the touchy issues, like AI ML is need of the hour. So ICMR came up with the ethical guidelines for AI. This has also been released. So this is the role we play when it comes to enabling policies, and we keep on doing that as per the requirements of the country. Regarding infrastructure development, we have discussed in the earlier slides that the requirement of the infrastructure is substantiated based on the requirements of the country. The funding support, I would really like to all of you to have a look at the kind of volumes of funding support that we give. Maybe we are not very good business development officers. We should reach out in a big way. Every year we have a national call wherein we give small grant of up to 2 CR, intermediate grant for doing multidisciplinary research and trials, multicentric studies of up to 8 CR, and centers of excellence up to 15 CR. So the centers of excellence, as I mentioned, should have interdisciplinary research, bunch of technologies which have alignment with the national health programs is what we would prefer to support. And we are now moving from basic research to translational research. So the projects which have translational potential leading to product development will be definitely preferred. Doing evidence-based research and disease surveillance is also one of our major activities. We maintain various kind of registries, be it a cancer registry, be it a COVID registry, because those are very important databases which can be used by the government, by the general people to look at and plan their studies. Preclinical and clinical study support infrastructure I have already talked with you all, and health technology assessment is also something exclusive that Department of Health Research does. And all of these are very important when it comes to medtech ecosystem. So these are some of the policies, as the Deputy Drug Controller also talked about, that bioethical guidelines, which we keep on revising every 2 to 3 years. Ethical guidelines for AI is something new, which we need to refer to. Yeah, they are all on our website. Yes, definitely. This policy I was talking about, entrepreneurship policy for medical professionals, it's also on our website. If you want, I can share all of them with the Ramji, and he can share with you all. So this is the policy I was talking about. We deliberately went to best of the people, I mean the minister, to launch this policy so that it becomes a national policy. Otherwise, making doctors work as entrepreneurs is really, really difficult. So this is now already there, and we have circulated it to all principal secretaries, principal health secretaries across the country. But of course, we need to really talk to people and see how well it is implemented. So that initial steps we have taken, but maybe we need to work more aggressively to ensure that it is implemented. I'll just give you 1 or 2 case studies on to just have an understanding on the kind of value addition we bring on board when it comes to devices and diagnostics. So one thing is to be kept in mind is that the technologies that we would like to support should have a larger footprint, potential to impact maximum population, and should be aligned with the national health programs. So this is a clear-cut example of trying to develop an automated AI platform for cervical cancer screening and diagnosis. We all know, I don't think that we need to appreciate and discuss the disease incidence of cervical cancer in our country. It's the largest impacting cancer among the women in our country, and it's again a global priority as well. We have around 2 crore people are screened every maybe in 2 to 3 years for cervical cancer screening. Current practice, we are aware we need to make a smear on a slide, and then we need to go to microscopically we have to see. It takes around 15-20 minutes even for a trained technician to come to a diagnosis that it is not a cancerous tissue or it is pre-cancerous, or to come to a conclusion. So it's a very skilled dependent technology. So we talked to these people at IIT Delhi, and now they have come out with this microscope which is called otoscope. This is going, and they also have an auto stain. So they have made a system for automatic staining of the making of the slides. 4 to 5 slides can be together screened with the help of otoscope. They needed a lot of data to train their AI tool. So there are some part of AI tool training has been done, but our institute NICPR, which is specializing in cancer, is working with them as a collaborator to give them authentic cases to positive, negative controls and the related cases, so as to train their AI tool. So now what will happen if it goes well, that auto staining will happen. So technicians will be able to auto stain the slide. 4 to 5 slides together will go to otoscope, and they will, with the help of AI tool, the device will be able to give at least screening decision on whether it is a pre-cancerous or it is a free case. If there is 1% doubt, it will go for further evaluation. So at least mass screening potential this kind of a device has. So we are supporting this device, and the way we are trying to help them is this M Pragati facility at IIT Delhi will help them with the prototype fabrication. Safdarjung Hospital is also on board as one of the sites, their microbiology lab, helping them with the validation part. And NICPR is their collaborator. We have made them do that. And then they were not sure about what is the category class wherein their device will fall. So we had a meeting with the regulator giving them clarity on the class wherein this device will fall based on the risk classification. And ISO 13485 training has been given to the team collectively at the IIT Delhi campus, wherein now they have clarity that from beginning they need to maintain. It's a startup driven project at IIT Delhi. They have to maintain files as per ISO 13485 documentation. So that's how from the beginning, if we identify that technology has potential, then things can be facilitated for such products, so that the translation can be accelerated. The data, yeah. The data is with ICMR. ICMR has an agreement with them. So all the data is taken by NICPR team. So particularly what ICMR is trying to do, but I can give you an example of TB diagnosis. We have a case on TB diagnosis. We have decided that among the ICMR system as directed by our DG, that there are 2 kind of data sets when it comes to AI tool. One is the testing set, and one is the testing set, and one is the validation set. So what the gold standard data set is of utmost importance when it comes to the development of AI tool. With the ministry is requesting ICMR that why don't you create the data set, which is the testing data set, and give it to put it in the public domain. So that we have done for TB. So you can go to our website, and 1 lakh data set for testing the tool, AI tool pertaining to TB is there on our website. So anybody can log in and take that data set. Likewise, we are trying to develop standard data set by we have created a center of excellence at ISC, wherein we are trying to create some gold standard data sets, which are authentic data sets for development of AI tools. So that's something interesting would be for training. Validation will happen through our institute, because validation should happen independently. So for training of the tool, testing data set will by and by we'll keep it in the public domain, and that the testing data set will retain with us, because that has to happen independently. So that data set will retain. So this is another very interesting example of developing. Ramji, I have time, or should I? 2-3 minutes. So I'll just give you one more example and then we'll close. Maybe we can have discussions later. This is again a very interesting example. We all know that India has to eliminate TB by 2025. World says, as per Sustainable Development Goals, world committed to end TB by 2030, but our country committed to do it by 2025. So we are working very, very hard to develop all kind of tools when it comes to TB diagnosis. We would also appreciate that patients generally do not come to diagnosis. If we have good diagnosis, then only we can treat. Treatment is possible. So we thought that why don't we, our team goes, some of maybe collaborators can go to the patient and do the screening. That mass screening is very much required, and for that we need very small tools. So this is the mandate which came from ICMR to the IITs, that please help us develop a portable handheld device for TB screening and diagnosis, because in our country we only have such tools developed by Korean organizations, and they are very, very costly. So if it comes to mass screening, we need a lot of tools, and they should be cost effective as well. Equally efficient, but cost effective. So we don't need to go more into the unmet need. We all know that the need is there. And now we are working hand in hand with the team at IIT Kanpur to develop this handheld X-ray device, wherein we have given them the testing data set to train their AI tool, looking at what are the requirements as far as BIS is concerned, what are the requirements of AERB. We are helping them to pass away the regulatory strategy, develop that in hand with them. And NIC and NIRT is our institute which is specializing in TB. We have made them as part of the team so that they can help them with their experience in expediting its development. So I think maybe the third example I can skip. I can just give you very briefly tell you that we all know that during COVID times, RT-PCR was the gold standard for doing the screening and diagnosis of COVID, and that would take 24 to 48 hours. So then our scientists at ICMR and we thought that we need to develop some RT-LAMP assay, which can give results in maybe 30 minutes to 1 hour, and that can be really helpful in doing, and that should also be cost effective. So this is another example of kind of technologies, the TB, this COVID-19 diagnostic kit that ICMR developed, looking at the unmet need of the country. And this was transferred to 2 companies. So I think these are some of the other technologies that we have validated. And in the end, I would just like to say that it's not only ICMR, particularly for medtech sector, all government agencies are working hand in hand to foster Make in India for the medical device sector. We are working for our country, not, we have, I think, cleared all interdepartmental barriers have been now crossed, and that's how we are here, coming to CSR and urging you to collaborate. Likewise, we are working with DST, with ICAR, and I think all of we at ICMR MDMS believe in working in consortia mode, and collaborations is the one by virtue of which we'll be able to achieve the big target of reducing that import dependency, all for public health. So this, with this, I would like to welcome you all to be part of our network, and we'll be happy to take technologies to the next level. Thank you very much.
Thank you so much, Dr. Suchita. We'd now like to open our platform for questions.
Excellent presentation, ma'am.
Thank you.
It is very sad that we have 80% devices. Way back in 2007-8, we worked on, I was in some other organization. I had worked on heart valves, dialysis machines.
Okay.
The trials being done in India, but it is being for the US FDA.
Okay.
Now, that is one question, that when will we see some therapeutic, all of it is all in vitro or medical diagnostics devices. The effort should be also on some therapy based devices, where, when will we see the Make in India heart valve or of course, stent, there is an initiative,
Right.
which Prime Minister's initiative is there. But the other things, I mean, there is a lot. I remember one device on stopping the kidney stone from coming down, and it is freely available in the US. And here we have hundreds, lakh crores of patients. So I'm just saying that, some efforts would be.
Yeah, definitely. I totally appreciate your point. And actually, the problems of medtech sector are, it's too much. It's too huge, and the sector is too diverse.
Yeah.
But since the time is limited, I have just told you about very easy technologies on which we are working, but we are also supporting LVAD stents, as you're mentioning. So we are doing some technology which are at the bottom of the pyramid, which we can immediately foster, and some high-end technologies also we are working on to look at in which we have huge import dependency.
Because diagnostics is not that big a challenge than, but therapy, yes, for a, say, example, a pacemaker, for example.
Yeah.
A patient who requires it for 10 years and spends lakhs and lakhs of
Yeah.
then everything is imported. One battery gets gone and then the whole.
To do it more systematically, in fact, we'll approach you all. We are going to do a very big landscape study so that we do things strategically, because the need of the sector is huge and too diverse. And most of the things are import dependent. So for us also to strategize on what are the things on which we should work first, and then second, and then third. With AMTZ, we are planning, and IITs, we are planning a big study, so that we move more systematically.
So one good thing is that you have a cross departmental barriers removed.
Yeah.
I would also recommend that, we could go across to the hub of the heart of medical device industry. That's in US. There are certain, there are 900 medical device companies in the US at this time, and they work. There may be some people, only 15 people in one company, but they are making millions of dollars of devices. So it's not really resource intensive. It can even be done from a garage shop, if for as all we know, for especially these things. So maybe there is a reach out to the country wise would really help to bring all those innovative technologies, which is no more. Many of them are no more innovations in those countries, but it would be for us.
Yeah, I think that's a very good suggestion.
Yeah,
but we are going step by step. Let's see if our bosses are inclined. We'll be happy to do anything.
And the second question was, you mentioned about that preclinical support.
Yeah.
What large animals are you talking about?
We are talking about dogs, goats, pigs.
Okay.
Sheep.
Not yet monkeys.
Monkeys is always there in NIV. We have in NARF also. Monkeys we have. Monkey studies we have in NARF.
For the pharma industry, most of us have to go abroad.
For NIV Institute has specialized facilities for doing monkey studies, and this NARF also has in Hyderabad. Yeah, actually there are some transit period is there. We had our director, some new director is going to come. So there are some technical challenges, but this facility is all set to go.
And this phase 1 facilities you mentioned, third point.
Phase 1 centers we have set up at 4 places.
I'm part of, we are working with one of them.
Okay, great.
The Chennai one is SRM Medical College.
I don't remember because some other team working on it.
No, it is SRM Medical College.
Okay.
We know we are
PGI, SRM, and KEM.
Yeah, so I'm part of some of those.
Okay,
things.
Great.
But then the center only has clinical facility. There's no bioanalytical at all. There's no bioanalytical at all. There's only clinical facility. There are beds, there are ICUs, but there's no. So even if a sponsor,
it will be great if you have some suggestions on because we are committed to strengthen their facilities. So we can help develop those.
I was a part of that SRM.
So that's great.
But then it did not happen.
Okay, so I can pass on your message.
Bioanalytical part, because that is the one biggest challenge. Because clinical
if we can get these suggestions in writing, we'll be happy to pass on this to the appropriate scientist who is looking at it.
Do you, I mean, the expertise level on various platforms is there in India. So ICMR, does it looking at any particular platform to promote our prioritized platforms? Because you can do PCR based, electrochemical or lateral flows. I mean, do you have any particular platform you want to put it above others, or is it that you want to treat
No,
others?
I think when it comes to problems of public health importance, all kind of technologies which work, government is ready to support. But of course, the ones which are standard of care and well acceptable in the market, for which acceptability is to and it's easy to operate by the people on the ground, because these technologies will be deployed at lowest places as well, like PHC, CHC, rural settings. So the question is that how easy those technologies are to be done, to be run by the people. So that skill development issue also we need to see, but we don't have any preference to some specific kind of technology set.
The reason, the understanding being in COVID time, everybody is familiar with PCR and the costs were brought down to 1/10 of what it was initially. So that's a kind of a big plus and maturity. So I thought, because that is also a strength, probably we can promote first compared to something else. That was a suggestion.
Yeah, I think every point needs a lot of deliberations to come to conclusion.
Quickly, last question quickly. Yeah. So it's not a question, but I would say that the thought that came while listening to you and correlating it with Dr. Ramakrishnan's presentation, right? The one challenge that I see for medical device is clinical validation, right? And if there is a process where and it is about collaboration within the industries as well. Suppose if as a company comes for clinical trial approach, right? And we know this that the endpoint that this clinical trial has match with a particular medical device that we have. Can we think of giving that some concessions to these companies to include these kind of medical devices or endpoints from these medical devices as an investigational endpoint rather than primary or secondary? Because companies might not like include their primary or secondary. But if the endpoints from these devices can be included as an investigational endpoint in those clinical trials, so that companies get benefits like for an orphan drug status, but as well as there is a validation that happens for these medical devices in clinical trials.
I think that's a good suggestion. Any other questions?
Thank you. Thank you, Dr.
Thank you.
Sujata, for a wonderful presentation. I would request Dr. Mathak to present a token of appreciation to Dr. Suchita. Thank you, Dr. Mathak. Thank you, Dr. Sujata. We are now moving on to our next session, and we are extremely delighted to have our next speaker, Mr. Ravi Kumar, who will be delivering his presentation on preclinical studies for NCEs. Mr. Ravi Kumar holds the position of Head of Toxicology at BioNeeds India Private Limited in Bangalore. With over 21 years of experience in the field of toxicology and an MPhil in life sciences, he brings a wealth of expertise to his role. Mr. Ravi Kumar has been a valuable part of the BioNeeds team since 2015 and currently leads the toxicology department. He oversees a diverse range of toxicology studies for both toxicology assessments and biocompatibility testing.
That should be. Very good morning to all. So I think most of the points were already covered by Dr. Ramakrishnan in the first presentation itself. So I was just wondering, what should I cover next? So I'll just do something like, in the first part, I'll just touch base with some of the initial studies that will be done before the clinical trials. In the next phase, I'll tell what all studies are required as a preclinical aspect, what all studies are required. And in the last phase, I'll just go through the alternate to animal studies, wherein that has been one of the important aspects in the preclinical studies now. So I'll be covering only this preclinical aspects because you can see a lot of drugs will go to the initial phase, and among them, very few go to the preclinical phase. And then when by the time they get into the clinical phase, there'll be one or two. That's all. So billions and billions of rupees are spent together, and then it takes about 10 to 12 years to complete the IND filing of one molecule. So I won't tell again and again because Dr. Ramakrishnan has told very briefly, very neatly about this preclinical studies and all. But however, the preclinical studies are important to have a safety of first-in-man studies because they give information on the efficacy aspects, the safety aspects, and then the eternity aspects also. The first-in-man studies. So the dose that will be taken from the NOEL point of view, from the animal dose to the human dose, there will be a 10 times factor. A safety factor of 10 times will be taken always. Except for, there are some exceptions only for some anti-cancer compounds where it has got a deleterious effects, or else there is a 10 times safety factor. Maybe the FDA accepts 1 time or 2 times, or else EU even accepts severely toxic dose 10, wherein 10% of the animals are allowed to die.
So the approaches that has been identified is like chemistry and proprietary. These are some of the tests that will be done in silico, in vivo, in the preclinical phase. Some studies are before the preclinical phases itself. See, main thing is the identification of the target and then the optimization to have to increase the affinity and then selectivity so that it will not have any deleterious effects. And then it's like potency or we call it as efficacy, and then metabolic stability so that it will increase the half-life of the compound. And then last is the bioavailability. See, that is the final thing when bioavailability needs to be increased for any kind of compounds. So initial studies that will be considered are the cytotoxicity tests, wherein it will be done on the living cells, so that whether we know that whether the test item has got the cytotoxic potential and all. One of the example is MTTSA. There is a calorimetric method, which has got the cancer cell lines, and it is done. It is based on the color changes. That's all. So membrane permeability. This is like you must have heard about the CACO2. So you won't go much into these because these are initial studies, and then these are the before the preclinical studies are conducted. This will be done on the CACO2 cell lines, wherein it is based on the diffusion method. But before that, the solubility has to be conducted, wherein the intestinal fluid will be used to know the solubility. And then protein binding or the CYP induction and CYP inhibition, this is called. These are basically used wherein the liver enzymes, the cytochrome P450 are involved in this, and it is majorly responsible for breaking down into active metabolites or inactive metabolites. Okay, harmful metabolites or useful metabolites. These are the things that happens here. So metabolic stability, this is also happens in the using the liver enzymes. Hepatic stability. So this is overview of preclinical studies. So here I'll be spending about a few time to explain you the study. But however, I won't go in depth into the guideline and all. Just I'll tell you the objective, the endpoint, or I'll just tell you what happens in the studies. Preclinical studies are differentiated into in vitro and in vivo. In vitro and cell lines and in vivo, there are pharmacology studies and toxicology studies. Pharmacology studies gives you efficacy, toxicology studies gives you safety, and then the starting dose will be evaluated. So it's like pharmacology studies, it's like we are going to induce the efficacy of the compound, whether the compound is efficacious or not, at what dose. So this is very important in this pharmacology. Yes, we just call it, it can be mouse model or anything. Suppose if it is an anti-cancer, please use good mice. If it is a diabetic, use Zucker rats, or what can I call you? So it depends. Selecting a relevant model is very much important to have this. Yes, I'll come again into the last slide. That's I'm just thinking of, because a lot of research has been going on using of alternate methods. I'll just tell what all studies needs to be connected. Then just I'll spend about, I won't take much time of yours because Dr. Ramakrishna, as I told you, he has covered most of the topic that I must have covered. Okay, in the battery of studies, genetic toxicology studies are the one. So 5 studies for IND submission, 5 studies are recommended, wherein 3 are in vitro and 2 are in vivo. Why along with the in vitro studies, in vivo recommend this? There might be false positive results, because in case of what you call the antibiotic, definitely there are chances of getting the false positive results. That's why in vivo. So the prediction includes all type, conclusion includes after completion of all types of studies. In case of Ames assay, just it's already mutated cell lines. So when you treat the animals, if the cell grows, then there is a problematic, because again the test item has got the capacity to revert the mutation. This is the endpoint. And then in vitro chromosomal aberration assay, in this we'll just check whether the chromosomes, whether there is any damage to the chromosomes after administration of test item. In vitro micronucleus test, so in the anaphase, it's like when the cell divides, there will be small, along with the nucleus, you can see a small, when you stain it, small dot kind of thing above the nucleus. So that's the micronucleus, means it does not divide completely. So that has got genotoxic potential. So these are all in vitro. For this in vitro studies, we have to look at during initial studies, there is no precipitation because precipitation means formation of layers will interrupt in reading of the colonies. And these are our mouse models. So mouse will be used, preferably Swiss Almond mouse will be used and then administered, and 12 to 16 hours after last administration, the bone marrow will be taken, harvested, and then read. In these two, human lymphocytes will be used in vitro studies. Mainly, this checks the damage to the chromosome genetic level and in turn the cancer. And then comes the general toxicology. This general toxicology, we need to choose a relevant model. One is a rodent and one is a round rodent. It's like in rodent, either rats or mouse, right? Either rat or mouse is chosen, and it needs to be an outbred state. Initially, the approach for this ion molecule is like, it cannot be administered like all other molecules. We need to have a lag period approach, means one dose to be administered, and there needs to be a lag period of about minimum of half an hour in case of intravenous, and in case of other routes, it can go up to 1 hour, 2 hours, 24 hours, even 48 hours also in case there is a delayed effect. So in this way, we need to increase the doses in rats, and then while increasing the doses, we need to set till what doses the animal tolerate or the mortality is noted. That's why it's called the maximum tolerated dose. Because since it's a pharma compound, we do not want a LD50 to be known. We want a maximum tolerated dose, minimum lethal dose, these kind of things. So based on that single dose study, MPD study, a small dose range finding study need to be scheduled. Using this dose range finding study, even a PK study can also be conducted as separately and as a part of this too. Our PK will be conducted just to know the kinetic profile of the compound. So what is the Tmax, what is the Cmax, what is the elimination rate, everything, this profile will be put upon. And then in this dose range finding study, it can be designed in such a way to extract maximum information, or it's just to fix the dose levels, because a single dose will not give you any information, and the long term, at least repeated dose administration will begin. However, this also needs to be planned, because all doses will not be administered daily. In case of anti-cancer, it will be based on cycles, or it will be once or twice a week. So these things are dosing regimen. It comes based on experience. That's all. Knowing the dosing regimen, the route of administration, the toxicologist designs this study. And until we complete the rat study, we do not get approval for the dog study, because large animal approval is not an easy task to get. And we need to submit the data of small molecules, small animals. And then MTD study in dogs. In this also, we need to administer single dose, wait for the animal to show clinical sign, and the same animal needs to be used in case of dogs, because we cannot use separate dogs, unlike small animals. We need to have either washout period and reuse, or wait for at least 3 to 4 days and administer the next dose. Yeah. And no, that comes in, that's like a lower species. That those are used in the like ecotoxicology, or you can use it as an alternate to animal studies to have some basic idea and all. Okay, then based on these data, we need to define a long-term study, whether it is a 7-day, 14-day, 28-day, or else if it is based on the cycles, anti-cancer compound, then a long-term toxic study needs to be scheduled, needs to be planned. Like if it in cycles once a week, twice a week, or it's daily administration. In this study, this will be as per the guideline with complete set of parameters, wherein the growth of the body weight, the feed consumption, the clinical signs, and then as a part of this, we can even evaluate the functional observation battery test also, and then the clinical pathology at the end of observation, and then the kinetics needs to be included, single administration kinetics, and then repeated administration kinetics at the end. If it is short term, first day and the last day, or if it is still long term, intermediate collection can also be planned. And then this gives a clear idea, the organ toxicity, systemic toxicity, because we need to have histopathology also along the part of the organ weight, gross changes. We need to cut open the animal and check for any gross changes. Yeah. Yes, it is all guideline driven. Guideline specifies that when you want to have this pivotal toxicity study, you need to include all the parameters. It's like a body weight, clinical signs, and at the end of treatment, means after completion of the treatment, you need to check whether the test item has brought in any local toxicity or systemic toxicity, or whether it is reversible or irreversible. All needs to be evaluated. So that's why we design it in such a way that after treatment, we collect the tissues, harvest it in formalin, and then we'll be having a recovery period also. We will be having a separate set of animals for those. Then we'll give a specified period of time, and after that, we do necropsy of those animals also and check whether the changes noted have reversed or not. It's like that. It's like first we need to complete rat study, and then the non-rodent, the dog study will be given approval. And next comes the safety pharmacology. Safety pharmacology studies as a part of IND filings, 3 studies are mandatory because as a part of pharmacodynamic effect, it should not bring in any effects on the physiological function of the animal. It's like as a CNS, they do the FOB, functional observation battery test, wherein the neuromuscular abnormalities will be checked, and which is also called as a Rat-Sirvins test. And then C is cardiovascular, wherein heart will be measured. Cardiovascular and respiratory, these 2 are in dog. Dogs will be used to do this activity, single dose, and then the parameters will be assessed. And then the next part is the developmental reproductive toxicology studies. Whether these studies need to be conducted or not before entering into phase 1. Up to here, these studies are sufficient. If the compound is given for ladies, if target is ladies, or woman, target or woman, then we need to conduct segment 1 studies, wherein it depicts the fertility studies. Or if there are any adverse effects noted in the reproductive organs during systemic toxicity or general toxicity study. At that time, we need to do this segment 1 study and then go into the phase 1. They'll give approval. Or else, if the compound that is in question, that is being tested, is being given to the women, childbearing ladies, then segment 2 also need to be done. Segment 2 in the sense, it's like we will be administering the test item to the pregnant rats or rabbits. No, not tropoblasts, it's the live animals. The animals. Yes, exactly. But we will not administer to the embryo, we are administered to the mother. It will pass the blood, yes, placenta, through the placenta. Because the administration pattern will be such that this is guideline driven from the day of implantation to the completion of organogenesis. That is from day 5 implantation. After fertilization on day 5, the fetus attaches itself to the wall of the uterus. It's called this one from the day of attachment to the completion of organogenesis, means closure of cleft palate. Up to day 19, administration will be done. And then before the animal deliver normally, the animal will be euthanized and cut open and the fetus will be taken. And then they will process for skeleton and visceral anomalies. Skeleton means in the sense the ossification, how the bone is formed. And then the visceral means how the tissues are formed, whether they are normal or abnormal. Yes, exactly, absolutely. We agree with that. So this needs to be done. So while doing this preclinical toxicology studies, some of the regulators even insist for alternate to animal studies. The main reason is, a lot of regulators now are emphasizing or insisting on 3 R's. That is reduction, refinement, and replacement. Reduction is reducing of number of animals. Refinement, just refine the procedure, whether you can refine the procedure so that the animal should not have more, it should not be in more pain or suffer or distress, or whether you can club 2 studies together to get the same objective. And then replacement, whether it can be replaced by an alternate to animal studies. And then the next is the zebrafish, what we are talking about. Yes. At least 5 to 6 animals are there. Yes, minimum 5 animals should be there to have a statistical analysis. 6 is ideal and can be submitted globally too. Because the Indian regulatory says 6 animals per sex per group. Per sex per group. Yes, 6 plus 6, 12 per group, because the toxicity needs to be evaluated in both sex. And then the OECD and all says 5 plus 5. So it goes on, the Red Book says similar, it also says the same thing. Long term, 10 plus 10. The number of animals depends upon the duration of treatment. The longer the duration, more the number of animals they will be keeping. Okay. So zebrafish, as an alternate to animal studies, zebrafish is one of the hot topic today because zebrafish is a linking factor between in vitro and in vivo. Because up to day 4, it's considered as an in vitro model. And from day 5 onwards, it's called an in vivo model because all the vital organs starts functioning. And the main reason is it can be easily seen through the microscope or the lens. And then one more, it shares about 70% of the genes with humans and 84% of the disease homologous with human beings. And also it has got a similar immune system like human beings, and hence a lot of research is being conducted on zebrafish, and then toxicity will be evaluated. And then apart from that, these epiderm and epialveolar, the 3-dimensional structure model, which are used for alternate to animal studies, are being used widely. This epiderm and epialveolar, which are from Matec Life Sciences, they supply global the 3-dimensional tissue models globally. And then epiderm is for alternate to dermal irritation, corrosion study, and epialveolar for inhalation study, single dose inhalation study. And then apart from that, human cell culture will be grown on a state of art culture. It's called as a organs on chips. These also can be used and then evaluated. And then stem cell research, a lot of research has been conducted, and one of the topmost company has conducted one of teratology study on stem cell research, and it is almost 95% accurate. Structural activity relationship, it's like a computer-based activity wherein the computer has got a program wherein based on the similar structure, it tells about the deleterious effects or adverse effects. So these are all the new technology that is being followed currently. These are all that is already globally. Some are relevant to preclinical studies and some are not. But however, these are followed in almost most of the CROs, offered as a standalone or as a separate study or as a part of studies required. Then finally, apart from the 3 R's that has been told, we also tell that relevance, responsibility, and respect has to be done when animal studies are being conducted. Because relevance, because choosing the right model is very much important. Responsibility, as a toxicologist, you need to report the data accurately and precisely, because one wrong thing that you do in the preclinical stage can bring lakhs, crores of loss to the owner or the owner of the chemical NCE. That's the reason you need to be more responsible. And then respect. Main thing is respect. Animals are being used as a model for preclinical research, and then even they also have the equal rights to live as you and me. Hence, while using them, we need to use them with respect and dignity. And last but not the least, I always tell that in toxicity study, all substances are poison. There is none that is not a poison. Only the right dose differentiates the poison from a remedy. Absolutely, sir. Absolutely. Yeah. Thank you, sir.
Thank you.
Yeah.
Thank you, Dr. Ravi. We now open our platform for any questions that our audiences might have. Any questions, anyone?
Sir, in silico trials, they'll be doing in a very initial stage. We need to have a computational based model that we need to have a specialization for that itself. That's a separate entity which starts in the very beginning stage of the IND in silico models. But it won't be so much accurate. Maybe you can tell that it's about 95 to 96% accurate, but not 100% accurate. No, actually, I'll tell you, maybe for a single dose, you must have an alternate to animal studies. But do we have an alternate to systemic toxicity? Will it be done for a 28-day or 90-day? Or do we have an alternate to reproductive toxicology study? No way. Still, see, regulators, one and here or there, they are accepting for single dose like that, but not for the systemic toxicity. Even now, the regulators are numb. Even now, they accept the studies wherein it is being conducted on animals. Yes, that is the as I told you, changes don't come overnight. We need to work on and on because we do not know what will happen. Because for a single dose, so many studies have come. Where was this single dose studies, alternate animal studies 10 years back? No. So we expect change during the coming years also. Maybe another 5 to 10 years, we will see that even for systemic toxicity or repetitive toxicity or carcinogenicity long term, we may have an alternate animal studies. Hope for the best, because there are a lot of toxicologists who might be on live or who might be in the podium also, who can just give the solution. Sorry. Cosmetics. See, to be frank, with cosmetics, testing of cosmetics on animals is banned long back. I think European testing of cosmetics on animals is banned, and only in vitro studies were accepted. And even in India now, studies, if you can go to the drug controller, studies on cosmetics is on animals is not accepted. Alternate animal studies are accepted. Yeah.
Any other questions?
Yeah. See, everything depends upon the therapeutic area and dosing regimen. Suppose if you have like feed additive, that feed additive doesn't have a limited period of time, then that needs to be taken for a longer duration, maybe 90 days or so and so, because it comes under Red Book. And some compounds are like therapeutic area, they have got a therapeutic duration, therapeutic route and therapeutic duration. So in that case, that much is sufficient, because after 1 week or 14 days period of time, the disease get vanished. That's all. See, suppose if you take a paracetamol for 3 to 5 days, you are sure that is sufficient. And for these kind of studies, especially deciding the duration of treatment depends upon the therapeutic indication and dosing regimen.
Thank you, Dr. Ravi. I would request Dr. Mathak to present a token of appreciation to Dr. Ravi. Can we have a round of applause for him? Thank you, Dr. Ravi. Now, I would like to take this opportunity to call upon Dr. Mathak. Dr. Kiran Mathak is currently designated as the Director of Medical and Regulatory Affairs at Veeda Clinical Research Limited. He is also a member of the board at Veeda. In this role, his responsibilities really encompass ensuring subject safety, protocol design, business development for new chemical entities, and overseeing clinical trials with NCs. With over 40 years of industrial experience, he excels in managing phase 1 studies, drug discovery and development, and international regulatory affairs, particularly related to drug discovery program. I would request Dr. Ramji to present a bouquet to Dr. Mathak before his presentation. Anvish, bouquet. Dr. Mathak will be speaking on subject safety in phase 1 trials. Thank you, Dr. Ramji. Dr. Mathak, you may begin your presentation.
One second. Yeah, so which one is for the next one? This is for the okay. No problem. So this thank you.
Well, good evening, and thank you very much for the still a good it's nearly 5:30 in the evening, and thank you very much. And thanks, Dr. Ramji and Dr. Madhusudan for allowing us to collaborate with them for this particular symposium, which is very useful, and I'm sure the startup companies will derive a lot of benefits out of it. because our organization has consciously decided to help the various startup companies, whether they are in the small molecule field or a large molecule field, or even in the medical devices. So if you have any issue, any problems, you can contact Dr. Hanji. He will give my email address and we'll help you out in even designing the clinical development program for you. So this is really to help so that we know the startup companies how they struggle. And certain time with due apologies, with a lack of knowledge can make you to come to a standstill. And or you may take some decisions which may not be desirable, or it may not be that economical. So we understand the value of startup companies and what is their need and how cost effective guidance they require. So with that, I will start with my talk on the clinical side. And I've taken a liberty of changing my topic because I knew Dr. Ram Kishan will talk more on the regulatory guidelines. So I changed the topic to more on the safety of the phase 1 studies. And so with that, I would like to move a little faster. My idea is to give you the aerial view of phase 1 studies, first in human studies, which is usually after the filing of an IND. Now, if you see here, the first block here, it is the initial application submission which is made, which is an IND application. And the review, you need to submit in the DCGI. And what I'm talking is essentially for the DCGI, that is Dutch Central Journal of India. And when we submit all the documents, if it is all in order as per the requirement, they take about at least 15 to 20 days to go through the documents. And then in case there is any query or anything, they would write to us, or the file is referred to the Subject Expert Committee. SEC stands for Subject Expert Committee. And these Subject Expert Committees are for various therapeutic areas. And they really you need to make a presentation to the Subject Expert Committee about your product, right from, if it is a first meeting, right from preclinical to the first in human being study that you are designing. So everything need to make presentation. Then you have many questions when you are making a presentation. So you need to be very careful in how you are making presentation, what presentation you are making, and justification for whatever studies you have carried out. So, and once the Subject Expert Committee gives an okay signal, then it takes about roughly another 2 to 3 weeks time to get the minutes of this Subject Expert Committee, and then the DCGI grants you the NOC, that is no objection certificate for conducting this first in human studies as such. So with that, you will realize that when you are doing a first in human study, the first time in human being, which is actually you are in a blind tunnel. You don't know what's going to happen. You have done animal studies, you have learned quite a lot based on the animal studies. But in human being, it can be entirely a different scenario. And therefore, there are a number of departments which has to work for when you are starting the first in human study. And that is the formulation department. They have to prepare a proper formulation. And of course, it has to be a GMP formulation. And I do know we had at least 2 startup companies who had a non-GMP formulation. And when they came to us and we had to decline that, sorry, we can't give this formulation to the human being. And their whole program went back by 2 years because when they had to remake the API which was GMP and then also make a formulation which is GMP, then do the stability data, etc. So their whole program delayed about 2 years, and thanks to somebody who gave them a wrong advice. Okay. Then there is a clinical development scientist, there is a clinical operations scientist, there is a toxicologist, as Dr. Ravi Kumar just mentioned here, and the clinical pharmacologist, safety physician, and regulatory affairs specialist. Now, safety physician is very important because the way the first in man study, the phase 1 study, is essentially to learn on the safety of the molecule. We are not evaluating the efficacy, but you are evaluating only the safety of the molecule at the various dosages. And so we learn about the safety of the molecule and then we can move forward. So safety is very important so far as the phase 1 studies are concerned. Now, like this, I would like to give an example of this monoclonal antibody. Sometimes in the year 2006, a German company which had prepared a monoclonal antibody as such, which if it had come in the market, it would have been good for the rheumatoid arthritis. And this study was conducted in one of the major hospitals in UK, in London. And one of the best units, nobody's fault, and lovely, desirable studies and the results in the preclinical data, which indicated that we can certainly move on with the first time in human being. And the ethics committee approval was granted based on the preclinical data and everything. And normally, for any phase 1 study, there are usually various cohorts. You have cohort 1 to cohort 5, where you increase the dose from cohort 1 to cohort 5 to learn about what is the safety profile at what doses. And this was the first cohort in which the drug was administered with the lowest possible dose. And when there were 8 subjects, there are 2 who received placebo and the 6 who received active drug. As soon as the drug is administered into 8 subjects, 6 who receive active drug, all of them develop anaphylactic shock. And they were all in ICU unit, and they were literally on the verge of deathbed. And out of these 6 subjects, there were at least 2 Indian students who were participating in the study. Also, the young subjects, you know. And those who took placebo did not develop anything, which indicated that the drug is responsible for causing an anaphylactic shock. And the best part, as I said, when the animal studies were carried out, there were no such reactions which was seen in the animals. And this is the species-wise variation which can take place in the first in human beings. So one has to be very careful. But fortunately, with God's grace, all 6 subjects who had anaphylactic shock, they all survived. And there was good luck on the part of the company and also on the part of the participants who participated in the study. But that led one thing, and that is early days before 2006. In UK, there was no room for taking an approval from the health authorities. It was considered as a more of an experimental pharmacology. And so this was done without obtaining the permission from MHRA. The ethics committee approval is a must. But subsequent to this, the regulatory authorities in UK made a rule that every first in human studies or phase 1 studies needs to have an approval from the regulatory authorities. They take about 30 to 35 days, but it has become now compulsory from 2006 onwards. So this is the kind of an incident that can take place in the first in human study. So what I'm telling you this is to make you understand that first in human study is not that easy to conduct. You know, it's very difficult. You have to have a good clinical knowledge, you need to have a good staff, you need to have good infrastructure, good analytical mind, what can happen, what cannot happen, signal detection, etc. You need to know about it.
Is there any way in the world where we are trying to do some pharmaceutical study before trying to?
Yes, you can do it, but it depends on the molecule. Molecule to molecule, it depends, and whether that particular molecule has any effect on the genes. So that you have to find out from the preclinical studies. Yeah. So there are various study design that one has to keep in mind for conducting the first in human studies. And these first in human studies can be carried out either in patients or in healthy subjects. In the patients, one has to do it if there is a lot of risk involved in the human being. Secondly, the target disease for which the molecule is being developed, whether that is how serious nature of the disease is, or there is any invasive kind of a administration of the drug. In healthy subjects, you can carry it out, and there one does not know, because what is happening is if you give it to the patient might be on other drugs also. Then you don't know what results that you see is because of any other concomitant drug administered or not. But in healthy subject, there is no other drugs being administered. So you know exactly whatever effect that you are seeing is because of the drug itself and nothing else. And this is the way to calculate how the drug is to be administered, what is the best dose to start with. And based on that is again, as Dr. Ravi Kumar mentioned about NOEL in the different animals. So based on that, you calculate the maximum dose which can be given to the healthy subjects or to the human beings. So that is nice to keep in mind. So based on that, you develop the human effective dose and also then the maximum recommended starting dose that you can calculate and then administer to the various healthy subjects or the patients. No. Here, what is important is the NOEL in the animals, because you are doing a first in human study. So it is based on whether it is in the rats or the dogs or the mice, and based on that there is a formula which you calculate, and then only you can calculate the dose, and then also you have to take the kind of a multiplication factor, the safety factor, and depending on again what you have seen in the animal study, whether there is a steep rise of the PK levels or whether there is kind of neurotherapeutic index or anything, then you have to be very careful in adjusting the dosages. So there is a different science entirely. I'm not going into too much detail because of the lack of time. There is also something like sentinel dosing. Now, sentinel dosing is such that, let's say, as I was mentioning, there are 8 subjects in a particular cohort. So that you don't want all 8 subjects, or out of 8, at least 6 subjects, like what happened in anaphylactic shock, in 6 subjects. So you don't want all 6 of them going into anaphylactic shock, and if you don't have proper infrastructure, you may not be able to treat them properly. And that could be calamity. So what now the people do is a sentinel dosing. And sentinel dosing, in that what they do is, out of 8, they will take only 2 subjects, one with active drug, one with placebo, and you treat them first. If you find everything is fine with it, there are no adverse effects, there is no toxicity, there is no life-threatening condition, etc., then you dose the remaining 6 subjects, in which there are 5 with active drug and 1 with placebo. And that is with every cohort, every dose that you escalate, whether it is a single ascending dose or whether it is a multiple ascending dose study. So that is how now what is preferred by most of the organizations, and also we recommend, is to do this sentinel dosing. So, and there are various trial design which are recommended, and one of the very common one is 3+3. This is again for the safety of the subjects. I'm not going into the details of it. There is also accelerated titration study design. There is also continuous evaluation Bayesian law design, which again very helpful to for conducting the studies. There is also a modified continuous reassessment method where you need to have a great help from the biostatistician who gives you an idea about how to move to the next higher dose and how to calculate the maximum tolerated dose. And also not only, and also the dose limiting toxicity, DLT, that you need to come to a conclusion with. There is also something like, which is very commonly now being preferred in the first in human being studies, is the combo studies, SAD, MAD combo studies. And that, if you look at this, the one is the ascending SAD dose, that is single ascending dose study, and in which case only one dose is administered to the subject. Okay, and that will keep on increasing the dose depending upon what the results that you see. Okay. Now, every now and then to get an approval. You must at least about 3 months in between. So what they do is, after a certain dose, you also need to understand what is the effect of the food on the formulation. So at a particular dose, you carry out a food effect study. The number of subjects remain the same. Almost every study which is done is always on the fasting stomach, empty stomach. And when you want to understand the food effect, one of the cohort who have already taken their medicine on empty stomach, after a short period, you administer the drug in the same subjects for the food. You have an effect in the pH levels of the drug on empty stomach and also on the after the food. Same way, again, in one of the cohort, every time actually all the study which are carried out are in healthy male subjects. And unless and until you have a reproductive toxicity study results available, you cannot administer the drug to the female subjects. But if the study is already carried out, the subject provided the reproductive toxicity study is safe. So in one of the cohort, what you can do is this, let's say this dose, you have got 8 subjects or 8 male subjects. With the same dose, again you dose only the female subjects. So you compare the gender, whether there is any difference in the pH levels between the male and the female genders as such. Okay. So all these kind of permutations and combinations which can be carried out. And that is why it is called as a combo studies, or is also known as an umbrella protocol, or is also called as the adaptive trial design. So you keep on adapting it. So that's an adaptive trial design. And when you do this phase 1 study, what you can come to know is that whether the drug at the various doses, whether it is proportional. What levels you see Cmax, AUC at let's say 10 milligram. When you increase to 20 milligram, whether the Cmax gets doubled or not, AUC gets doubled or not. If you go to 40 milligram, then again it's doubled than 20. So you come to or whether it is not dose proportional. If it is not proportional, then you don't know how it is going to end up with. You cannot predict the adverse events. But if it is dose proportional, if you see that 20 milligram something, with 40 milligram, you expect it to see much more than what you have seen at 20 milligram, because the people will get doubled with that. Okay, so this is the kind of thing that one has to understand whether it is dose proportional or not. Also, this is, I've just learned this, but I have seen the effect in one of the studies that I was doing. In female subjects, the levels were much higher than the male subjects. And the levels were as high as 4 times higher of Cmax and 5 times higher of AUC in female subjects compared to male subjects. And then the further study, we need to see that we reduce in female subjects, because the levels which I saw with 40 milligram was same as what I would see in 200 milligram dose. So that is why we have to reduce the dose. And this is the food effect study. Again, the study that I conducted. This is on empty stomach. And this is the level that you see after the food of that particular molecule study that I carried out. And also the various formulations, it can give you an idea of whether the immediate release or with the delayed release or the prolonged release, or the how it will affect the tolerability profile of the molecule. And this gives you an idea of see the same dose administered to 5 subjects and see the levels in these subjects. And that shows there is something wrong with the formulation. And you need to stop at this level and rethink about whether you need to redesign the formulation or not. So this is again the study which I have carried out in my unit. Yeah. And of course, safety pharmacology, Dr. Ravi already mentioned, so I'm not going into that. And what is very important is the safety considerations every time that we have to keep in mind as such. And look at the dosing that you have to select whether it is appropriate or not. whether the dose escalation is proper or not. You can have 10, 20, 40, 80, but you can't have 10 and then you jump straight to 100. So there is a way of doing it. So that is the way one has to do it, the direction of the drug administration. Then there is monitoring that one has to carry out so that you understand what is happening to the subjects. In the protocol design, you need to identify when to stop the study. The study going up to 1000 milligram, but you're seeing something at 500 milligram, which is based on whether it's the Cmax, Tmax, AUC, adverse effect profile, etc. You need to put it down when you stop the study as such, and that is the guideline which is required. And for every phase 1 study, there has to be a data safety monitoring board. With every cohort results, they have to evaluate whether you can go to the next higher dose or not. But that's an independent body who gives an idea about whether you can go or you cannot go forward. There are various challenges which I mentioned in the phase 1 studies. I'm not going to the details of it. And so phase 1 study, as you see, is a very important aspect in the development of the molecule. The safety of the subjects is the most important aspect which you have to keep in mind. You cannot compromise safety of the subject so far as the phase 1 study or any study is concerned. Right? Thank you very much for your patient hearing.
Thank you, Dr. Mathur. So we are open for any Q&A. If someone has any question, we can just proceed.
I'm sorry, I'm asking them not to ask more questions because my colleagues, they have to take a flight to go to Ahmedabad. So that's the reason. So they need to reach in time to the airport.
So
I think we have one more speaker.
Yes.
Yeah, thank you. Thank you very much for your patience. I enjoyed it.
So now we are coming to our last session on analytical methods for assessing high order structure in biosimilars. And I would like to call upon Dr. Ravi Bhardwaj. Dr. Ravi serves as the head of analytics and characterization in Veeda Clinical Research in Biopharma division in Bangalore. In this pivotal role, he oversees biopharmaceutical operations with a focus of structural characterization of therapeutic agents driven by a passion for innovation and technology. He leverages his extensive experience from renowned companies like Novozymes, Biopharma, Waters, Dr. Reddy's, and many more. So I will now call Dr. Mathur to have present the memento for Dr. Rajiv.
In case you want to use this, it's on.
Yeah, sure. Thank you very much.
Thank you, Dr. Rajiv. Over to you.
Greetings to the dignitaries and members of this symposium. Thank you for the patience. We'll be switching gears and moving towards characterization or analytical perspective of the biotherapeutics. So basically, the focus would be to look at higher order structure and its importance in biotherapeutics. So I would like to basically look at what is HOS, how does it play an important role in biotherapeutics, and some of the regulators' perspective in HOS, and also the methods available which we can leverage for the biotherapeutics. So if you would like to introduce to HOS, so the basic definition would be, if you look at biopharmaceuticals, mostly they are majorly of protein in nature. So monoclonal antibodies, ADCs, fusion proteins, such like. So the question comes here is that they are complex in their structure. Then further, when it moves towards various development strategies, for example, there could be a change in the process, how we manufacture the protein or develop a particular protein. There could also be a change in the product. Probably you can make some changes to the product to fit the clinical conditions, and also the formulation in which the product. So these changes basically encounter to a situation where we have a native structure of the protein, and when we change all these parameters, what is the correct structure? So how do we move out and what happens if there are some changes in the structure. So that is where the HOS comes into picture. And if I compare both a biologic versus a biosimilar, the pathways for biologics is mostly discovery driven and development driven. However, look at the biosimilar, it's mostly a development heavy workflows. However, either of the case we work with, the structure and conformation of the molecules are very important. Why? Because it has a direct impact on the function of the molecule. That's where the end product is destined to be, and this in fact determines the quality, safety, stability, and efficacy of the drug. So the higher order structure concept comes in where we look at the structure and conformation in detail, and that is one of the critical quality attribute for the biopharmaceuticals. And if you look at what is the factor, why do we really need to look into the higher order structure, would be look at the size of the molecule. It's 150 kilodalton, right? And look at the expression systems. It is basically expressed in the mammalian systems, and it has a lot of pathways in which a lot of modifications can be induced to these molecules. And associated with a lot of post-translational modification, a lot of generation of variants, and our favorite cysteines present on the protein gives rise to a lot of disulfide linkages. So looking at all these complexities, basically there are two factors which determines, or I would call influencers in determining the higher order structure. So those two are the molecular folding and the 3D conformation of the protein. So with this concept, I would like to move further. Looking at, we did discuss about the importance of the structure and its direct influence on the function. We do look at two stages. One is at the primary structure, we call it physicochemical characterization. The other would be the higher order structure, where we look at mostly on the secondary, tertiary, and quaternary structures. And why is it important? Biosimilars, because we have to monitor the same parameters in batch-to-batch consistency, and also look at the stability of the drug, as well as address or monitor these variants and aggregates at various stages, as I said, in the process, in the product, or in the formulation. Well, the structure looks very easy in terms of image, but if you look at the 3D structure, it's a very complex structure made up of a lot of alpha helices, beta sheets, and a lot of components which make this structure. And the important concept in terms of biosimilars is two major objectives. Biosimilars being a development-heavy workflow in biosimilars, we aim to demonstrate the comparability of the drug with the innovator. So that is basically the objective. And with this, we move to a typical development pathway in a biosimilar for higher order structure monitoring. So if you look at once the project has been initiated, we have developments at different phases in terms of process development, analytical, and formulation. And they are post the development activity, the product quality assessment is basically destined for POC in terms of in vivo studies. And there's a parallel activity which happens for characterization of this higher order structure. And post characterization, we come to an evaluation stage. So that is where we compare whether the structure what we intended is similar post this process optimization, and also evaluate whether the higher order structure profiles have been impacted by any of these variants or aggregates or any kind of modifications. The next stage would be, we should look at whether do we need changes. That is basically a decision point where we say, what is the HOS profile versus in vivo data. Based on this, the scientist takes a decision about, do we really look into redevelopment of the process in case there are changes? If no, then we are good to go forward with the toxicology studies. So with this, I would also come to factors which are important for higher order structure. So basically, we look at a lot of stability of the molecule. That stability can correlate in terms of its conformation, the structure, its function stability, thermal, and also how good the molecule is not aggregating and how good the molecule is solubilizing. So the various factors which basically in part, its influence is basically, if I can take an example of oxidation, you can see it has effects on almost all the stability parameters, and also it can cause aggregation to the proteins. Similarly, you have glycosylation, deamidation, pH, temperature, and ion exchange. So all these basically are the influences which can change the stability of the molecule and also gives rise to aggregation and solubility related concerns, which are basically important for any molecule to have a structural integrity of same. And looking at the regulator's perspective, yes, we do have a very defined, statement in ICH guidelines about higher order structure and its importance in the product, as well as what methods should be leveraged to compare the higher order structure in terms of a biosimilar. And one of the regulators also explicitly mentions that higher order structure is very critical in terms of determining your biosimilarity. So again, the methods needs to be very concise. It should be robust enough to monitor your batch-to-batch characterization and so on and so forth. So that's about the regulator's perspective. And moving on to the methodologies and expertise, we basically monitor, sorry for the text, looks like there's some, yeah. Some studies which can be done at the initial when the drug is approved, and later on for a robust monitoring, there are a different set of studies So we have a battery of tests for HOS, and with that we pick few of them, which is used for routine monitoring of the batches. But once you're finished with the development, you need to execute all the methods for the product, which you intend to push it to the next stage.
First variation in terms of the initial set, which we want to do for approval of the drug, and there would be a different set of experiments with less cost, which will be used for a batch to batch.
Exactly, yeah, similarly. So I have a list of methods, probably I can correlate in that.
Thank you.
Moving on to what methods we have to employ versus what we look at. So there are four important things which we look at. One is the secondary structure, which consists of alpha helix, beta sheets, and trans and loop. Tertiary structure is one level above the secondary structure, where you have a lot of interactions such as ionic interaction, disulfide bonds, and various other interactions. Quaternary is when the protein converts itself into a multi-protein complex. It could be a dimer, trimer, which is functionally active. And yes. And protein conformants are which you come across when you have these process development and the product characterization stages. So just to summarize the methods which we have, this is the list of exhaustive methods for higher order structure. It starts from hydrogen deuterium exchange to ion mobility to CD, Raman, NMR, X-ray. So there's endless methods which you can employ basically to study all these measurements. And if I have to correlate this particular methods with its, what effect it has or what kind of measurements we do, I can take one of the example here. If you look at the circular dichroism spectrum, it's one of a measurement technique which basically measures the secondary structure and the tertiary structure of the protein, depending on what kind of UV range we have, so far UV and near UV. So that's how the measurement happens. And most important methods such as CD, FTIR, DAC, DLS, ML, MALS, all these contribute to a lot of measurements coming out from the secondary, tertiary, and quaternary structure. And some methods require, downstream monitoring. So when you're purifying your protein, you basically have to make sure there are no conformers or any stability-related concerns. So for that, we have a lot of methods such as dynamic light scattering and multi-angle light scattering. Yes, we have picked up some robust methods which are required for method development. Probably next slide I can take you there. So this is the expertise we have at Veeda. The objective, the rationale behind picking up these methods, where basically we want to ensure we have suitable methods for an early screening, as well as have a full characterization methods and also routine monitoring. So these 5 methods are operational at Veeda. And looking at the instruments, we have CD from Applied Photophysics, FTI from Bruker, SEC MALS from PostNova, and DAC DLS from TA and Anton Parr. In terms of expertise, we do have worked on a lot of MABs, you can name them, and also peptides, recombinant proteins, fusion proteins, and a lot of CGT products have been also done. And the takeaway message is these all are compliance-driven methods. So these methods can be fully developed, qualified, and also validated. So that's the take-home from the methods. And when we look at the biopharma solutions at Veeda, the HOS part is part of an integral workflow. However, we do have robust methods for primary structure, where we heavily bank on the high-resolution mass spectrometry, followed by capillary electrophoresis, where we study exclusively on the primary structure of the protein, right from its molecular weight to the sequence, and also all other attributes which are associated with the primary structure. HOS, we've already discussed. It's about secondary and tertiary structure mainly, and also the conformers which can be generated during our development process. And apart from that, we do have analytical support for which includes HPLC and high throughput methods, basically to look at charge variance or size variant and any other associated impurities which we come across during the development. So this is the summary of the methods in terms of HOS as well as other solutions which we offer at Veeda. So just a glimpse about some of the high resolution and high throughput technologies. So these are the high resolution mass spectrometers and also as microcapillary based high throughput technologies, where you can see the rationale behind having a high resolution and a high throughput is basically you don't require to spend too much time when you're looking multiple samples. So we have to cut down on the runtime. Whereas when we are investigating or characterizing a particular molecule, we do need to go into the high resolution aspects. So it's a fine balance between what do we need to look at and what we expect the outcome from. With this, I would like to summarize my discussion.
Thank you, Dr. Rajiv. We would like to open our platform for any questions that our audiences might have.
Yes, for mRNA, we have methods, something on AEX, anion exchange chromatography. We have capillary electrophoresis, LIF-based, a fluorescence-based method for an mRNA characterization. So these two are already proven methods for mRNA. Not from India, we have seen some global companies coming up with these mRNA products, which have been successful. Yeah.
Any other questions?
I know yours says clinical research, but is there any example of food proteins or anything being characterized using the similar methodologies? So there's this full domain where the alternative food proteins are coming up. So I was wondering, I understand that the regulatory this thing is a little bit less for food alternative proteins, but how many of these would be required for any idea in that sense?
Well, the protein which comes out on post purification, the characterization has to undergo the listed methods here. So my PhD work was also on a food protein. It's an alpha amylase inhibitor from one of the food source. So but again, the complexity or the structural fold, I mean, it depends on what kind of protein, I mean, how complex the higher order structure is. So if it is only limited to the secondary structure, we can use very basic methods such as the CD or FTIR.
Like something post-translational modification, PTMs and all being there. So then what would be the best method for characterization and all those things?
If you're looking at PTM, then peptide mapping would be a better method. And also we do have multi-attribute method, which is basically an advanced version of peptide mapping, where we look at multiple PTMs in one method.
Okay.
So, yeah.
Thank you.
Thank you so much, Dr. Raji, for a wonderful presentation. I would like to sincerely thank all the speakers for their insightful presentations. Since we have come to an end of this wonderful symposium, it's time to express our gratitude and appreciation. I would like to invite Dr. Kiran Mathur to deliver the vote of thanks.
Thank you, Ragi. And I have I'm the 11th planning to come in the field now. And that's one of the big things that I would like to do is to thank everybody who made this is very helpful and at the same time, we need to understand that this kind of program cannot happen with a very good team, dedicated team, and hardworking colleagues. And I would like to thank our chief guest, Dr. Ramakrishnan. And I know he is very busy. And he said, sure, I will come. And without the invitation, he just accepted the invitation when I met him. And I think that's something very good. And we do know that the last 2 days, the US FDA was here. And I also was there in the meeting with him. But we have participated in IDM meetings, so. But in spite of that, we came in and I think he deserves a big round of applause for coming over and giving us really good knowledge. If I want to put it from the horse's mouth, you know, and he really gives some good practical examples, how to go about doing various things, and also to give importance on the ethics and other things, which is, I think, very important as well, and consequences which you do not follow. So that's something very good, and I think I'm working for the Dr. Ramakrishnan for coming over. Another one is from CCMB team, and I applied Dr. Ranji and then to Dr. Madhusudhan, very accepted the idea of having this particular symposium. And the amount of cooperation, the amount of enthusiasm which is being shown is amazing. And so I'm thankful to Dr. Ramji, Dr. Madhusudhan. The state is excellent, and what that you are seeing is due to her and also to Meghna. So I cannot forget Meghna, who is also very helpful. I can see I was here from 11 o'clock, she was running around and trying to see the things happen the way it should be. So I think that CCMB, Meghna, Ritika, Dr. Madhusudhan, and Dr. Ranji, we are indeed thankful to you. And we will be to collaborate like this in future also for the benefit of the startup companies. Of course, we had a very good speakers, Dr. Jagannath from Novartis, Dr. Suchit from ICMR, Dr. Rajiv Bhatia, we just presented from Veeda Clinical, Dr. Veeda Clinical from BioPharmas and Atal Incubation. And all the arrangements and all the background work we done for a young people, we are saying we done very very down, and they now from the marketing and the business development. And very efficient, from pillar to post for getting things done. I must compliment them for doing an excellent job, and I must give them a round of applause. And in the team, I must give out the names and the Mr. Rajkumar Agarwal, who is head of business development. He is seated here in the second row. And I think it deserves a round of applause because in spite of a busy schedule, we spent right from 12 o'clock till now in this particular venue is something very good. And I know we keep on traveling all over the world. This was very important. Setting an example colleagues in the additional advantage. So as well, I think he not yet is in the back office. Show who is in the back office. He was there outside taking the names, so he has come down from Bangalore to and all of these people except Mr. Rajkumar, they all are from outside of Hyderabad. So which is the name that I read out now. We have seen her in the pink suit. She has come from there. So you can imagine that from the meeting, you're going back to it. So simply, she is very good. Then we are in the back office. Anisha was short but and lifting a very, very thing and the mementos and the flowers and everything. So thank you, Anisha. Wonderful, wonderful. And I cannot forget Mr. Siddharth Bard. He's head of corporate communication, and he came over specifically for this meeting, and we had put him in charge to take care of Dr. Ramkishan, so that nothing goes wrong, because we have to take care of everybody. So, and of course, I cannot forget the audience. Without the audience, this would have been absolutely a hopeless and a failure program. So a big round of applause for the audience who have participated in a big number. So thank you very much, each and every participant who have taken care and participated here in this meeting. And I must also thank our vendors who have helped us in organizing various materials here. And that is Webstream Communication Private Limited and the Homes Kitchen, who have served us a lovely food in this particular space. So thank you very much once again for them also. With that, I call it a day, and thank you very much, and hope to see you all again. Just keep in mind, if you have any questions, any comments, anything that you need, you can write to us. We are there to help you. We can hold your hand. No problems at all. Okay. And I want to end with an apology for the projection, which was not that very visible, but I believe we have done a recording. So whoever has participated, we have their email ID. I'm sure our marketing team will do the editing and other things, and they'll send you a link to see, so that you can actually see the slides that is being projected. So thank you very much once again. Okay, bye-bye.